quality. You cannot expect not any lag. And actually, if I'm not wrong, OBS imposes a 3-second delay.
The picture quality looks good.
Okay. Okay, great. Seems to be working. Seems to be working properly right now.
And don't forget that you are seeing the preview. You are not seeing the I don't know if you are doing the preview or really going live and checking.
Now it's really going live.
It's really going live. Don't worry, if the quality is good, if the image quality is good, the sound quality is good.
Yeah.
Here's about the delay. Nobody is seeing that from the room.
Right, they'll just see the whole. They'll just see the whole video, but like a few seconds after what we see, right? It won't be it'll be continuous, but just a little behind, right?
Yeah.
Okay, then I'm fine with that. I'm fine with that.
For people watching it, they have no idea that it's delayed.
Right.
Only when they will text you, you will have like a little bit to answer. Okay. That's just a detail.
Okay.
Is it working out?
Yes.
Okay. That's good.
Okay.
No.
Oh, wait.
We have the same error.
It was working. Now the same error came up.
You have internet connection, yeah.
Yes.
And is the quality bad?
So it's just like a yellow banner saying the 15 frame frequency of 4 seconds on that. Did a YouTube say?
But is the quality of the video for you as bad?
I think it looks good, right? But it's just slow, right?
I mean, if that just means it's further behind, I'm okay with that. I mean,
If the stream quality is okay, check the virtual cam the whole view to see if it's coming down. There's some tools, virtual cam, check, see if the right button is highlighted.
Okay, virtual cam, there's a few checkboxes, like auto start, horizontal flip,
okay.
or keep aspect ratio.
So cancel virtual cam.
Hello?
Cancel all virtual cam.
Yeah, it's all canceled. Then there's a target. There's a target, it's OBS camera, and there's a
I do.
delay frame of 4.
I do. To edit it. I just don't know how to. I want to add the link for the YouTube
Left one.
that we've done.
The right. On the right.
Right is called P out, right?
Yes.
So the right one, the right button should be. So actually, click on the left one or click on the right one.
Okay.
Click on the right one.
Click on the right one. That means okay. So the right button will be. Then do I need to change the delay frame?
Need to change the delay frame.
Yeah, do I have to change the delay frame?
On that guy? That's supposed to stop.
Yeah. So Joel was telling us that it's he felt that YouTube was easier. We could do we could do comments on YouTube, or we could have them send the comments on Facebook. I don't know. Do they want to try to keep the comments private or
Okay. Try to.
It needs to stop virtual cam.
Sure. Okay. Yeah.
How to stop the virtual cam?
And you can just get my YouTube, and they can have a choice.
So just deactivate and activate.
Deactivate all virtual cam.
Okay. All right.
Virtual cam on OBS, right? That's what you're doing.
Okay. Thank you.
So sometimes that's what starts. It's 3 or 9.
Okay. John, we're about to start. So when you come here, you come upstairs and take a look.
Yeah. It starts at 9, right?
Yes. No, it started like 84, right? We're starting now.
84. You are trying out?
Yeah, I'm starting now. So whenever you're here, please come upstairs.
Yeah, I'll check on it. Okay.
Okay.
Yeah.
Thank you.
Okay. So there's a post on the Facebook page.
Now I just not worry about the middle part. Find the channel.
Himanshu, yeah, he's great.
So,
Yeah.
Right, and could you tell me that 9 minutes?
I can.
Sure. I just mean like just give them like some sign.
Raise your hand.
Yeah, like raise your hand at 9 minutes and then
And then 12 I think just
and then yeah.
Falling on keywords.
Very bad.
The chair will remind you, it's 9 minutes or 12 minutes. 9 minutes.
around 9, 10 minutes.
Okay, that's fine. I was just like, I didn't know if I only had 10 minutes.
No, if you have 12 minutes,
12 minutes.
Okay.
Can you just say like few words? I didn't pick something up on the ground floor. Just like say, I think you all.
Thank you guys for coming for the 2019 CPLC and CBQB students and postdoc symposium, and welcome. So we have 3 sessions for the morning, and we have like 2 coffee breaks, and then we have lunch after all the 3 sessions. And for each session we have a chair, that is we have 3 volunteers, and the chair will introduce the speaker and keep the length of the presentation to be only 10 to 12 minutes, and we have 3 minutes or 5 minutes for questions. And let's get started.
Good morning all. On behalf of the organizers, I would like to welcome you all in the CPLC Symposium. And our first speaker for the symposium is Kevin Rand. And Kevin is from SUA Mang Group. And he's going to talk about the fuzz mutations in fuzz which are related to these neurological diseases. And he'll talk about the phase transition in fuzz proteins. Kevin.
Thank you. Thank you to the organizers for inviting me here, and I'm excited to talk about my research involving this protein called FUS, which is implicated in both ALS and FTLD, which are neurodegenerative diseases. And so FUS exists in both the cytoplasm and the nucleus of your cells, and in ALS patients, they have both a wild-type copy and a mutant copy. These versions of FUS can undergo RNA-mediated oligomerization. So RNA is used as a scaffold in order to form condensates of FUS that undergo liquid-liquid phase separation. In the nucleus, FUS is more diffuse because there's a higher RNA concentration, and in the nucleus, FUS is involved in splicing, mRNA processing, and the damage response. There's this protein called carioferrin beta 2, which is nuclearly important, that can bring FUS and can break apart FUS aggregates and import it into the nucleus. However, in ALS mutations, this process is disrupted by these mutations that prevent this movement into the nucleus. So, because ALS patients are heterozygous, I wanted to ask this question of whether the wild-type version of FUS can help recover some of the defects that we see with these mutant versions of FUS. And so a lot of the questions that I am trying to answer involve are looking at this recovery effect and the interaction between wild type and mutant FUS. So in our recent paper that we just published, we classified FUS into we classified ALS mutations of FUS into 2 different categories. The first is mutations in glycine. Mutations in glycine cause solid-like aggregates to form, which have a more solid-like properties than liquid-like phase separation usually happens with FUS. By contrast, these arginine mutations cause much larger condensates to form. So you have much larger droplets forming both in cells and in vivo. And then these interactions are likely due, or these changes in the FUS condensate properties are likely due, or we showed that they are due to changes in the RNA binding properties of these FUS mutations. And so there is some limited recovery of these mutations by karyogram beta 2. However, I wanted to see if wild type could recover these mutations to more of a wild type like behavior in my work. And so the first question I wanted to ask is whether ALS FUS mutants interact with wild type FUS. And so this seems like kind of a silly question, like how could a point mutation in FUS cause it to no longer interact? But it's motivated by this recent paper that found that even just changing the RNA binding partner of this protein called WEE3 can cause these immiscible droplets to form when you color code the RNAs. So you can see here that using these 2 different RNAs causes these droplets to form that have separate identities. And so I wanted to answer whether wild type and mutant FUS interact by doing essentially the same experiment that was shown here, but with FUS, by doing a single molecule assay in which I nucleate FUS on RNA at the single molecule level. And then I'm starting to do some in vivo work by co-transfecting 2 plasmids of FUS into neuroblastoma cells. And so first I looked at these arginine mutations. And so again, arginine mutants are aggregate prone and they form these large condensates. When I label wild type FUS with Cy3, you can see I form these circular droplets that remain circular over time. And they also remain circular if I label with Cy5. If I combine the 2 proteins in an equimolar ratio, I have complete colocalization of the 2 different wild type FUS, which is what I would expect. And then with arginine mutations, so these point mutations in arginine that are linked with ALS, R244C, R216C, and R521G, what I see is also complete colocalization between wild type and the arginine mutations. And so I wanted to then test our other class mutations. So glycine mutations form these solid-like aggregates, as I mentioned before. And really strikingly, what I found is that when you add the Cy5 and Cy3 versions, or the Cy5 glycine mutations to the Cy3 wild type, you get immiscible droplets to form, no matter where the glycine mutation is located. So these are throughout the FUS disorder domains. And what you see here is that there are these immiscible droplets forming. And it's no matter where it happens with all these different glycine mutations. I do want to note that they start to interact more as time goes on further. And I'm not exactly sure why that's happening yet. So if I quantify this by just taking the number of yellow droplets and dividing it by the total number of droplets, I see that there's a drastic decrease in the number of colocalized or yellow droplets of these glycine mutations. And so I wanted to see if this phenotype was conserved at the single molecule level. And so I developed an assay in which we have a single molecule surface with an unlabeled RNA. This RNA has a 50 uracil stretch that I can that FUS likes to bind to. And if I flow in Cy3 and Cy5 FUS, I can watch it bind to the RNA in real time. And so I'm going to show you hypothetical data for immiscible FUS and an immiscible FUS interaction. So immiscible FUS, you might see both proteins bind in both the red excitation, which is tracking the Cy5 binding, and the green excitation, which is tracking the Cy3 binding. And I'm going to process this data into these heat maps here. And so this trace right here, which is one RNA that has 2 red proteins bind and 1 green protein bind, gets sorted into this bin right here. And so that I did that for a few hundred traces, and then by contrast, an immiscible FUS would show something more like this, in which if you look at the red excitation, you'd see only the red excitation protein binding, but nothing in the green excitation. And in the heat map, that would show as more density on the outside of the heat map as opposed to the inside. And so at the single molecule level, or on our slides, basically we just flow in FUS, and it's about to start to float in. These bright spots are FUS binding to RNA on our single molecule surface. And so wild type, we see some density in the middle of the heat map. There's a lot of monomers forming, which I don't really count because they can't possibly be mixed. But when you look at the arginine mutations, you see a lot of density in the middle of the heat map, and this indicates that the proteins seem to be mixing at the single molecule level. There's no preference for one to bind by itself. And so naturally I looked at the glycine mutations, and by contrast you see a lot of density on the outside of the heat map. And this is because there's only one protein binding, and once that protein binds, it seems to self-select for other proteins of that same mutation. If I quantify this, I see a moderate decrease in the colocalized, or the number of colocalized traces for these glycine mutations, which is really encouraging. And so next I'm starting to look at these results in cells to see if I can link what I've seen in vitro with what I've seen in vivo. And so these are neuroblastoma cell lines, and what I'm showing you here is a wild-type GFP plasmid and a wild-type haloplasmid. And in unstressed conditions, the FUS is diffused throughout the cytoplasmic nucleus. As I stress these cells with sodium arsenite, these punctae form, which are basically droplets inside of cells. And then if I co-transfect with these two plasmids together, I can get yellow aggregates to form with the cell. And so naturally I want to try this with both the wild-type and the mutants, but I haven't gotten to that yet. And so just to check in so far, what I've shown you so far is that if you multimerize glycine with RNA, or glycine mutations with RNA and wild type, you'll see miscible droplets start to form. This seems like a logical mechanism for glycine mutations to achieve ALS or FTD progression, in which you have this glycine mutation that has its own aggregate forming separate of wild type. It's probably pathogenic and can drive ALS progression. By contrast, if you have this mixed interaction, there might be some sort of rescue. And so I wanted to test for this effect by using RNA as the output. And so in the Myung lab, we've developed this assay in which we have, instead of an unlabeled RNA, we have a FRET-labeled RNA and unlabeled FUS. As we flow in FUS, we see FRET pair changes between the FRET pairs on the RNA. So if you have RNA alone, that has a low FRET efficiency. But when you flow in the wild-type version of FUS, you get these rapid FRET fluctuations that go away in these ALS mutations. These ALS mutations lack these FRET fluctuations. And so I can use this as an output to see how the FUS is interacting with RNA. And so what I did is I started with a moderate concentration of the mutants by themselves, and I titrated in more of the wild-type. And what I was looking for was an increase in the FRET fluctuations. And so with the arginine mutations, I see a pretty drastic increase in FRET fluctuations, whereas with the glycine mutations, there's not as rapid of a reemergence of these FRET fluctuations. And if I quantify that here with these violin plots, where the bottom of the violin plot is traces with fewer FRET fluctuations, and the top is more FRET fluctuations, I see a rapid reemergence of these FRET fluctuations with arginine, but not with glycine, which was really encouraging, and seems like this wild-type arginine interaction might be ameliorating some of the defects that you see with arginine with FUS. I also want to look at droplets again, but instead of using labeled FUS as my output, I'm using labeled RNA. So I can have these Side3 labeled U40 stretches that form these droplets here, as you see. And the important thing about using the fluorescent RNA is I can do fluorescence recovery after photobleaching. And so I can bleach this droplet here and then watch the fluorescence reemerge as RNA exchanges into the droplet. And so when I have wild-type droplets form and R244C droplets form, you can see that visually they're very different. R244C droplets are much larger. When I mix them together in an equal molar ratio, I find that the droplets return to their wild-type size, which is really exciting and backs up what I'm seeing at the single molecule level. If I quantify this here, you can see that R244C forms really large droplets, whereas this mixed population is closer to wild-type. If I zoom into this time point here, You can see I'm basically just losing all these really large droplets if I plot every single droplet on this dot plot. So I'm really losing these large droplets tend to form with UR244C mutation. By contrast, the G156E mutation forms droplets that are visually very similar to wild type, so I look at the fluorescence recovery after photobleaching. And what I see is that there's an intermediate fluorescence recovery for the mixed population in between the G mutation and the wild type. This is actually because I'm averaging out the immiscible droplets. So I have a few wild type droplets I test and a few glycine droplets I test, and if you average them out, you get something in the middle of the two of them. For some reason, this difference goes away over time, and this might be due to the increased interaction. And so this leads to a potential pathogenesis mechanism in which if you have a more miscible droplet, there might be more recovery, whereas a more immiscible droplet might show no recovery. And what I've shown you so far is that mutant FUS physically interacts with wild type FUS, whereas R mutant FUS physically interacts with wild type FUS, whereas G mutant does not. R mutants can be rescued by wild type FUS, whereas G mutants cannot. And then this rescue effect is exhibited at multiple scales, including at the molecule level and at the ensemble level. And with that, I'd like to thank my lab, especially Sua, for being an excellent mentor and for really pushing me to grow as a scientist, and our funding sources and my students who helped drive this research. I'll take questions.
For questions? I'm sorry. Okay, so we'll move on.
I have a question.
Alec.
So what do you think happens there?
Can you repeat the question?
The question was like, how does it come about from work?
So at the molecular, it's hard to do structural studies on FUS because it's disordered and doesn't crystallize, or you can't really use cryo-EM and other things like that. But it's probably some sort of defect in the oligomerization of FUS on RNA that's causing these problems, where the mutations in glycines affect the actual folding of these ultimeric interactions. But we don't know for sure.
Okay, if there are no more questions, thanks to Kevin for nice talk. So it's time for our second speaker. It's Mayank Wu from Torres Pogorelia and Martin Goble group. And Mayank is going to talk about how does small osmolytes protect protein from feeling the heat. And Mayank, you have 12 minutes before we start taking the questions.
So hello everyone, I'm Mayank. Hopefully the coffee has kicked in by now. So today I'm going to be talking about something that I've been doing for the past 2 years. So our work in the lab involves a lot of understanding of how protein folding is affected by different factors that are present in the cell. So the factors that you can think that are affecting the cell can be thought of as pH, cell-free volume, pressure, temperature, chemotropic, cosmotropic agents, which can generally be known as osmolytes. So the project that I'm going to be talking about focuses on chemotropic, cosmotropic agents and temperature, and how some osmolytes can rescue proteins from being denatured in the presence of at high temperatures. So osmolytes are like small molecules that are present in the cell and can be expressed as the total number of solutes present in the cell. And the osmolarity can be expressed as total number of solutes present per liter. So these small molecules are very important for maintaining the overall fluid balance and cell viscosity, and can have overall a vital role in different cellular functions. In particular, they can stabilize or destabilize proteins. So just to give you an example of how they can stabilize or destabilize proteins. So what can happen is that the proteins might not like to interact with the folded state, but however they might not like to interact with the unfolded state even more, resulting in a net stabilization effect. So the osmolite that in particular that I'm interested in studying is TMAO. So TMAO is trimethylamine oxide. It has been shown to be a potent stabilizer for proteins. This is the structure for TMAO, and as you can see that there is a partial charge that's present on the N and the O, and also there is like a pretty significant hydrophilic group, giving rise to hydrophobic and hydrophilic natures of the osmolite. So, in particular, TMAO stabilizes proteins against pressure and chemical denaturation. It has traditionally been used to counteract the effects of urea. As you go down to the depths of the ocean, the concentration of TMAO almost increases linearly in fishes, which is interesting to note because that's at a huge depth. That's almost like 8,000 meters. The amount of pressure that most fishes experience is almost 11,000 times the pressure that we are experiencing right now. So the model protein that I'm studying is protein B. It is a small 3-alpha helical bundle, and it is from the albumin binding domain of from the bacterial surface protein PAB. So this protein has been optimized to fold very fast, 4 microseconds, which makes it ideal for simulation studies because now we have been able to reach the microsecond mark pretty normally. So that helps us to be able to capture a number of folded and unfolded events for this protein. And as you can see, this protein is also rich in hydrophobic and hydrophilic regions, and thus its interaction with TMAO is interesting to understand. So the simulation details that I used for my for this project are as follows. We used Chromax. The water model that was used was TIP3P. We simulated almost 192 microseconds of 12 different conditions. So the 12 different conditions were at 3 different concentrations of TMAO and 3 different concentrations of TMAO and 4 different temperatures. We used the CHARM 36 force field, and in particular we used the CAST force field for TMAO. So because TMAO has this dual nature, it has been harder to characterize its force field. There are a bunch of force fields present out there in literature. I started out with 3 different force fields and then narrowed down onto the CAST force field because that was the most widely accepted, and we couldn't see any particular differences between the 3 force fields that we used, at least for the parameters that was used. So this is how the graph looks like. So here I have plotted the Q, which is the fraction of native contacts, which can be just thought of as an indicator of whether the protein is folded or unfolded. So in particular, if it is 1, it is folded, and if it is nearer to 0, it's unfolded. And we have given it the gap as 0.8 to 1 to be declared as folded ensemble, the region between 0.2 to 0.8 as the intermediate ensemble, and 0 to 0.2 as to be the unfolded ensemble. So as you can see, as you go on increasing the temperature, the amount of unfolded population increases. This is something to be expected as there is more thermal energy present in the system. Secondly, as you go on increasing the TMAO concentration, the amount of folded population increases. So this seems to, so previously a lot of studies have been done against the pressure and chemical rescue from TMAO, but in particular this is for a significantly large protein, which is like almost 47 amino acids. This is the first time that temperature stabilization has been shown. So the reason for this that we have think is something similar to that is related to the pressure denaturation. So here I have plotted the radial distribution function of TMAO and water around the folded ensembles or the different of the protein, and as you can see, the TMAO is excluded from the hydration shell of the protein. So as you go on from the protein surface, first you get a large peak where you can see the first hydration shell, then you get a TMAO shell, and then there is an almost alternating TMAO hydration shell where it then flattens out to the bulk density. So what we see in particular is that in the folded ensemble, water is pushed in by TMAO. So as you can see here in water, where there is no TMAO present, the radial distribution function is increases to almost 2.5 in when TMAO is present. So what we think is happening is that TMAO does not like to interact with the protein itself, but is pushing in water, which is helping it keep it stable and keep it protecting it from getting unfolded. So the other thing that we wanted to see was how does the void volume of the protein change, because that is the major factor that can lead to pressure unfolding, and TMAO generally protects proteins against pressure unfolding. So here I have plotted the normal solvent accessible surface area versus the fraction of void volume, and as you can see, even though the protein is getting unfolded, the change in the void volume is much lower when in the presence of TMAO. So just to summarize the project, so TMAO stabilizes proteins from temperature denaturation. TMAO does not seem to interact directly with the water or protein. Water is pushed in towards by protein in the presence of TMAO, and the void volume of the protein remains more relatively constant even when the protein gets unfolded. So the future directions that I want to study are, instead of using like a small protein, to use slightly larger protein and conduct pressure melts from organism at high depth, and to simulate pressure unfolding for this large protein in the presence of TMAO, and to determine this particular role that TMAO seems to be playing against pressure denaturation. And currently I have been working on finalizing the sequence to be used for this protein. So in particular, we have identified 3 sequences from 3 different angler fishes, and that's what we have been working on. And just to acknowledge, I would like to thank my group and my advisors and the funding sources.
Thanks, man. Questions?
Okay, so 2 questions. So one is that the concentration 1 molar and 2 molar TMO. Did you have any aggregation during the simulation between TMO? No. Then how did you measure or calculate void?
So we used a program called Volcan. So
and these are constant pressure simulations, constant volume?
These are constant pressure simulations.
So what is causing void?
So the voids that are present in the protein itself.
Oh, I see.
So that can open up here.
Thank you.
So this gas force, what is it?
So cache force field is more or less similar to the CHARM-MM parameterization that is done by FFTK. So it is so there are modifications to the cache force field, but the cache force field that we used are just like quantum mechanical calculations done and then setting the parameters, like optimizing the parameters for CHARM-MM. But there are some force fields that optimize in particular, just the partial charges present on NNO with regards to different. So there was another force field that we used called NETS force field. So that optimized it for activity coefficient of TMO in different solutions.
But does it bother you that it's not CHARM? Because you're using CHARM for protein.
No, so that's what, yeah. So the cache force field is closest to CHARM.
And if you just use CHARM, what happens?
CHARM, I don't think CHARM has any parameters directly for. So I use CGenFF. So CGenFF and CAST match almost completely. So that's why, because CAST was the more widely used, we use CAST.
I have one question.
Yeah.
So, what kind of ion concentration do you use?
So the ion concentration was around 20 micromolar, 20 millimolar. Sorry.
Okay. And where do you see these ions binding to the protein? Because you can see that in the geophile.
I have not looked into it, but I can.
Sure. Well, if no further questions, thanks, Mayank, for excellent talk. And new speaker is Ryan from my chain group, and Ryan is going to talk about multifunctional platform for studying neurotransmitter release from single neurons. So you have a Oh, okay.
Did you have the mic?
Mike,
Did you use
do you have a mic?
Oh, sorry.
Hello everyone. Today I'm going to talk about the multifunctional platform for studying neurotransmitter release from single neurons. So, electrophysiology has widely been used in the study of neurotransmission, and it can generate information about the neuronal activities and how the neurons communicate with each other. And however, another way to look at neurotransmission is to detect and quantify the neurotransmitter being released using chemical measurements, including HPLC, mass spec, electrochemistry, and so on. And among the techniques, electrochemistry serves as a very powerful platform to study the neurotransmission from single cells in the living cell condition. And this will be the focus of my talk today. So traditionally, to detect neurotransmitters, a carbon electrode is being used, and based on the oxidation and reduction of the neurotransmitters, these redox-active neurotransmitters can be detected, such as dopamine, serotonin, phenylphosphine, octopamine, and so on. And on the other hand, there are many neurotransmitters that's not redox-active, such as GABA, acetylcholine, and so on. So it is, they cannot be detected on carbon fibers, so carbon electrodes. So we have to develop new electrodes for the detection of these species. So our group, so this will be the first part of my talk. So our group has detected the acetylcholine, have developed the acetylcholine detection electrode using ITIES electrodes. It is the short form of interface between two immeasurable electrolyte solutions. So in short, our probe is a 4-glass pipette filled with oil phase, and this probe is positioned into an aqueous phase. So at the very end of the tip, this interface between two immeasurable electrolyte solutions is formed. And when we apply a negative potential in the oil phase, it is going to attract positive ions into the probe, and we detect the ion transfer using so this transfer gives the ion transfer current, and this is how we detect the ions. And because acetylcholine is positively charged, this is how we detect acetylcholine. And once we have the probe, the next step is to find a way to bring the probe very close to a single cell. So this we developed. We use a technique called scanning electron microscopy, in short SECM. It has nanometer resolution
in terms of spatial resolution. And so scanning electron microscopy is the system where you can position the probe very close to a sample surface. And once you are very close to the sample surface, you can do surface imaging, you can study the fast electron transfer kinetics, or you can study the neurotransmitter release from the sample. And it has widely been used in biological research and energy research. And so once we have our probe and the technique to position the probe near the single cells, we have recently published reported the detection of acetylcholine release from single neurons. So the animal model we studied is a C-slug, aplysia. We took the individual neurons from the pedal ganglion and cultured it on the silicon wafer. And this is how, so we try to study the position of the probe far from the neuron, and upon stimulation, we did not really see any detection signals. However, when we position the probe like 140 nanometer above the neuron, upon chemical stimulation, a clear release signal was detected. And now we try to take one step further and try to develop this multifunctional platform that can simultaneously detect both acetylcholine release and cell firing. So this is how experimental setup looks like. This is the nanopipette, and we use it, the nanoprobe. We use it for the acetylcholine detection. And this is electrophysiology probe, and it is used for intracellular recording of action potentials. And finally, we have this stimulating pipette that is to apply chemical stimulation. So with this setup, we are able to detect the acetylcholine release and the cell firing at the same time. So the dashed line indicates the application of chemical stimulation, and we observe cell firing, which indicates the vesicle release events. And shortly after that, our chemical probe detected the signal corresponding to acetylcholine release. So from this chemical detection, we generate quantitative information about what's the concentration being released, what's the length, duration of the event, and how many molecules are being released. And also from this action potential recording, we can calculate what is the cell firing rate. And also, as we studied, we've also detected the non-cholinergic neurons. So these neurons did fire action potential upon chemical stimulation. However, no cholinergic transmitter was being observed. And this is consistent with the results that shows not every cell inside the pedal ganglion is cholinergic. So as a short summary, so simultaneous action potential and the cholinergic transmitter release has been measured on the single cell. And monitoring these two parameters at the same time can provide a better understanding of the process of neurotransmission. And our results open doors to study transmission from a variety of neural models. So for the second part of my talk, I will talk about our recent effort to develop carbon nanoelectrodes that enables the detection of redox-active neurotransmitters such as dopamine, serotonin, and octopamine. So we first pull our glass pipette using a laser puller. Then we deposit it with carbon through pyrolysis. As you can see, after pyrolysis, the pipette is filled with carbon, yet we do not have well-controlled depend and with well. So we do not have well control of the shape and the geometry. So we take this pipette and use focused ion beam to mill the tip end, and this gives us a flat surface with controllable geometry. Next, we characterize these carbon nanoelectrodes using electrochemical measurements. So this is cyclic voltammetry, which shows the carbon electrode has good chemical reactivity, and this tip current corresponded well with the tip size measured from SEM images. And then we confirm the tip has a disc shape and a flat surface. So we position the tip, we approach the tip close to the substrate using scanning electrochemical microscopy. And when the tip gets close to the substrate, the current on the tip will decrease because of the hindered diffusion towards the tip. And this behavior can be described by a theoretical equation. So we fitted our experimental results, which are the blue data, with this theoretical curve, which is the red one, and we can see that it fits very well. So this means that our carbon electrode behaves very well according to the theory that actually describes the disc-shaped electrode. And also, from this theoretical curve, we can calculate this endpoint, what is the distance. So we calculated that, and it is within 100 nanometers towards the silicon wafer surface. So this confirms that our tip has has a flat surface as well. So then we use this carbon electrode and use it to detect dopamine, serotonin, octopamine based on the oxidation. And as this is the cyclic of the oxidation of the 3 neurotransmitters, and you can see that the 3 neurotransmitters are oxidized at different potentials. So this is dopamine, serotonin, and octopamine. So this gives us the opportunity to distinguish them in amperometry studies. For example, if the solution only contains dopamine, when we apply potential at 0.2 volt, we will see signal. 0.4 volt, we will see signal, and 0.6, 0.85, we will all see signals. And if the solution contains serotonin, there will be not a signal. I mean, there won't be a signal at 0.2 volt, but 0.2. But we will see signals when the tip is biased at 0.4, 0.6, and 0.85 volts. And similarly, when we have octopamine, we will not see anything at 0.4, 0.2, or 0.4 volt, but we will see it at 0.6 and 0.85 volts. And with this, we use scanning electron microscopy, bring our probe very close to the neuron, and apply chemical stimulation, and this is the result we observed. So when we bias tip at 0.2 volt, we do not see any detection. When we bias the tip at 0.4 volt, yeah, no detection. And the 0.6 and 0.85, we do see detection. So this means dopamine is being released from the single neuron. And as a summary, we have developed this carbon nanoelectrode for the detection of dopamine release from single neurons. And this carbon electrode can also be used to study the transmission of dopamine, serotonin, and other neuron models. And we will develop this multifunctional platform to detect these redox active species and monitor the cell firing simultaneously from other types of neuron models. And I would like to thank Professor Shen for the opportunity for me to participate in this project and also for the mentoring of the project. I'd like to thank our undergrad assistants, Jeremy for helping me capturing the aplysia neurons, Daniel for helping me preparing the carbon electrodes, and Teresa, Kristen, Michelle for the work on the single cell release study. And I was our collaborator and our funding sources.
Thank you.
So what you do is you hold the electrode very close to the surface,
Yes.
but you're not puncturing at all.
Not puncturing because of this curve. We can stop. But, so the approach curve is operated by a piezo. So we set the parameters to tell it to stop when it hits, say, 40% of the current. So it stops there. Yeah.
So I had actually a similar question to Paul. So this is, you have 140 micron distance between your electrode and the neuron?
40 nanometers.
Nanometers?
Yes.
Okay. Okay.
Because so this is relative D over A. So yeah. So if you have smaller probe, you actually achieve much smaller distance.
Is this the closest you can get to the neuron?
So we can push forward, but we have a chance of hitting. So we don't know when. So we don't know until it hits. When it hits, this curve will deviate from the standard.
Yeah.
Yes. So we want to be conservative.
Then can I ask one more
Yeah.
question? So your ITIES method, I mean, is it any charged molecule is detected at the interface, or does it have to be? Why do you only see acetylcholine and not?
So it's based on the potential we apply. So different molecules enter the interface at different potentials, basically because of the depending on the hydrophobicity of the molecule.
The amphiphilic.
Yeah.
Gotcha.
Yeah.
What is the temporal resolution?
Temporal resolution depending on our potential stats. So right now our temporal resolution is maybe 16 milliseconds. But if we use a better potential stat like the multiclam something, yeah, multiclam 700, those can give us maybe 100 microsecond resolution. Yes, just we haven't done it yet.
What is your sensitivity? I guess in terms of molecules.
Sensitivity. So something like micromolar. So So the limit of detection, lowest limit of detection, depending on the tip size, can be something like 10 micromolar or 100 micromolar if we use even smaller tips. So if we have smaller tips, it's about 100 micromolar. But if we use a bit larger tip, we get something like 10, 20 micromolar. Yeah.
Very good question. So, how small in your hands can your filler be?
So this carbon one, I can make something like 200 nanometers so far. But if the ITIES, really depending on the fuller, so I can make as small as 15 nanometers, or reliably like 200, 300 nanometers.
Yeah, that's right. Because my colleagues, they use it to actually puncture.
Yes. That's another issue. Like when the tips are too small, sometimes I can enter the cell without telling it from the approach curve. Maybe just open hole on the cell membrane, just enters it without any feedback.
Okay, one more question. So let's say you're approaching a cell surface and you're interested in knowing, are there acetylcholine receptors or GABA receptors or AMPA receptors? Is there a way that you can differentiate between the various receptor types?
We are studying it's a good question. We haven't thought about that actually, because we are studying the release. So we're not really detecting what happens on the receptors. But let's say we have two neurons grown together, and maybe we can stimulate the first cell and see what the second one does, but that's something I have not thought about.
Yeah, if there are no more questions, let's thank Rand. The last speaker before the coffee break is Karna Gaura from Cefecon Lab Group. And Karna is going to talk about predicting microbial community metabolic functions for genomic structure. Karna, thank you.
Okay, thank you everybody. I'm happy to be here to talk about my postdoctoral work in the lab of Cefecon in the Department of Physics. So the functioning and persistence of ecosystems depends on the continual cycling of nutrients. And the cycling is performed in many cases by microbial communities. So, for example, in the global cycling of nitrogen, bacteria and other microorganisms fix dinitrogen in the atmosphere to ammonia, which is a bioavailable form of nitrogen. Nitrifying bacteria convert ammonia to nitrate, and then denitrifiers convert nitrate back into dinitrogen gas through a sequence of reduction reactions that go through several intermediates. So the focus of my work that I'll tell you about today is about characterizing quantitatively the process of denitrification. So in particular ecosystems, nutrient cycling is not typically done by individual organisms, but by whole communities. In the specific case of denitrification, within a community, one might find a mix of organisms which do different steps in the process. So in this sense, nutrient cycling is a product of the emergent metabolism of a number of interacting populations. So the question that we're asking is, can we predict metabolite flows from genomes? So the genomes of organisms in a community encode the functional capabilities, the enzymes, In principle, they also encode information about how organisms respond to environmental cues and interact with other populations. So the question is there information, sufficient information in genomes to predict the flows of metabolites at the community level? And we've chosen to study denitrification in part because the sort of parts list, so to speak, of the process genomically is well characterized. So the reductase enzymes are known, as are some of the regulatory structure and the transporters. So the question is, can we use this information to predict community metabolism? So the approach that we're taking in this project is a sort of top-down statistical empirical approach. First, we isolate denitrifying bacteria from local soils. And the point here is to construct a genomically diverse ensemble of bacteria. We construct communities in the lab and culture them in the lab and make metabolite measurements. So we'll focus on making measurements of nitrate and nitrite, which are easy for us to do in high throughput. Also, we will do whole genome sequencing of all of the isolates in our collection, characterize their genomic content, specifically their denitrifying capabilities with respect to what denitrifying genes they have, and use the variation in these genomes to predict the observed metabolite dynamics that we measure in the lab. Okay, so first we isolated and characterized a library of 64 strains. So these are bacterial isolates from local soils. We observed 3 major phenotypes. So I'll call these NAR-NER, NAR, and NER. Okay, so NAR-NER strains are capable of doing the first 2 steps of the process. NAR strains do only the first, and NER strains do only the second. And indeed, this library of strains is diverse. We have representatives from both alpha, beta, and gamma proteobacteria classes. And I'd like to thank some collaborators for helping with the isolation process. Next, we performed measurements of these strains in monoculture. So these plots are example metabolite flows for each of these major phenotypes. So for example, here, I'm showing the dynamics of a NAR-NER strain. So initially I supply it with some nitrate. It consumes that nitrate and thereby producing some nitrite. The nitrite accumulates to a certain level and then later gets consumed. And that goes to nitric oxide, which we don't measure. For a NAR strain, you see the initially supplied nitrate diminishing, and in proportion the amount of nitrite increases. And for a NER strain, you see just the nitrite decreasing. Our first result is that a simple consumer resource model does a good job of describing the denitrification dynamics of the strains in our library. So you can think, so the model comprises 3 parts: population and a variable representing the dynamics of nitrate and nitrite. So to first approximation, the population is growing exponentially, which is at a rate proportional to the nitrate reduction rate of the strain. So this is the model for a nitrate reducer. The consumption of nitrate, or the reduction of nitrate, is in proportion to the population size. And the accumulation of nitrite is in a 1-to-1 stoichiometry with the reduction of nitrate. We use endpoint optical density measurements to estimate a yield parameter. And we then use nonlinear optimization to fit the model, the remaining parameter, the reduction rate, to the observed dynamics of the single strain. So thereby we obtain a quantitative phenotype, parameterizing the strain with a reduction rate and a yield. Our second result is that community dynamics, by and large, are predicted from monocultures. So here I'm showing an example for when you put 2 NAR strains together. So strain I and strain J. Individually, we obtain parameters parameterizations of each of these strains, which are represented by RA sub I, RAI, gamma AI, RAJ, gamma AJ. We can extend the consumer resource model via simply adding the rate contributions of each strain together to form an additive model prediction for what happens when you put these two strains together. So in this prediction, we assume essentially only resource competition is driving the interaction between these two strains. What we observe in most measured communities of two strains is that the two-strain additive prediction does an excellent job of fitting the observed dynamics. So what this means in essence is that the problem of predicting community metabolism is reduced or simplified somewhat. So in order to predict the dynamics of a community, one needs only to predict the dynamics of monocultures. So in order to predict single strain dynamics from genomes, we formulated a simple regression problem where we relate the phenotypic parameters to the presence or absence of denitrification genes that each strain possesses. So here is for a preliminary data set of a handful of strains, the presence-absence matrix of denitrification-related genes. So these genes are the nitrate reductases, the nitrite reductases, nitric oxide, nitrous oxide. This is the variation in the regulatory content. These are transporters associated with denitrification. So the objective here is, or the sort of assumption of our regression approach is that the presence or absence of a gene has some additive contribution to the quantitative value of a phenotypic parameter. And using L1 regularized regression, we obtain a sparse solution to this regression problem. So essentially, a couple of genes are able to quantitatively predict the nitrate reduction rate in this example that I'm showing, and similar regressions apply to the other parameters that we characterize. So this result shows that a simple characterization of the genomic content of our bacterial communities is able to predict metabolic dynamics at the community level. And right now we're working to expand this result to our larger library of strains. I'd like to thank my postdoctoral mentors, Seth Kuhn in the Department of Physics and Madhav Mani at Northwestern Applied Math, and Derek Peng and Laura Troyer for helping with this project. Thank you. Questions? Yes.
What do you think it tells us that the genome actually, just looking at the genome actually succeeds so well in predicting? So should I think of it as meaning that regulation is sort of perfect, it's done whenever it's needed, or there's not much regulation? How should I think about it?
Yeah, so I mean, if you examine this particular example, there is one regulatory element which exerts a strong positive influence on the reduction rate in this case. But I'm not sure exactly how to interpret this. I mean, so one interpretation might be that this gene is mechanistically related to an increased reduction rate. But I think that we're very far from being able to say that. Another interpretation is that this gene is in some way correlated with the gene that is mechanistically related to an increase in reduction rate. So I'm not sure how to answer that question at this point.
Well, if there are no questions, thanks Karna once again. And we'll stop here for a coffee break for 15 minutes and we'll get back to the further talks at the 10. Thanks.
Oh yeah, it's very easy to walk around.
Yeah, go ahead.
Yeah.
Yeah, but go ahead.
Do I need to watch all of these? Or
Pardon me?
do I need to watch the rest? Or is it okay? Yeah, but
Yeah, it's really up to you.
I need to. I mean, I have cells, so I need to study
Yeah,
them.
then just,
He wants to do it.
He wants to do
Where? I should check here?
Yeah. You can try first. You can try first. Oh, yeah. He's
yeah, I probably have so probably have a
yeah.
Yeah, so it's
Not on the wall. Not on the wall.
I don't know.
I don't. You can try.
Yeah, so my background is
Try both.
no.
Yeah, but this is
PD analysis. You should check it.
Well, so they enable, you know.
Hey, you do the simulations. Yeah.
One molar die.
So the right thing to do. Not so much. I mean, numerical simulation. Okay, that's good. The scale is much smaller than that. I can run it on my laptop.
So this connector is only here.
Yeah.
You should,
This is your.
I guess you said that.
No, I have a. Okay.
Yeah, just be close to it.
Okay.
Oh, yeah. So that's this file.
Are you saying that you had trouble? Sure. Oh, okay. Yeah, that's cool. Yeah.
Increase the volume very fast.
Which
So either you use the solution.
And I'll try to increase the log back.
Yeah. Master.
yeah, this first is
It's the numbers.
you can actually
I haven't yet, although I did.
I twist myself like that. I'm almost better. Try to figure out.
Oh, turn it off. We can give it to the chair and they can hand it to the. We do that.
That might help.
So basically, once I told you yesterday. But it also messes. It's why you have to kill it. Already extra. Just for like small things. That's why it's kind of difficult to start.
Sorry, are you the first talker in this?
No, I'm just checking.
That's fine.
Yeah, ask today.
I'm second last.
Second to last. Of the whole thing or?
Of all the things.
Because he has a vision. Yeah, I wrote him up several times, but previously we were writing, we'll see. I mean, I said it's like, oh, I know, but it's funny, it was it's the way the University of Illinois job search page. There you go. Click and click and click and click, and then you find the actual it's so difficult to
Okay, first
other people, you know, you see people add enough place, put it, put stuff
Yes. Okay. If you do,
so I was able to find it, but
well, yeah.
but the link that we have on our
And all been done.
So you're just it's recently at home. Yeah, I wasn't sure if it was
Oh, yeah. Find a back.
Better not knock over these.
It's probably carbon
我也觉得我以后就我说我也觉得我以后Yeah, actually, you're right. Yeah. And now I'm not.
Yeah, everyone, including the Q&A. Sheldon, I'm going to remind them. No, remind that 9 and 12.
From my side, please.
我听说有一个孩子follower 出去。Like?
Yeah, that's what I was thinking. I was like, oh, it's possible.
Also,
Yeah.
I had asked a few years ago if you
You have to leave early. I asked him to get Okay, yeah. Is someone offering help?
No, I've been there before. Yeah, for summer school.
Okay, all right. Okay. Well, if you need some help, talk to me or someone else or something.
Well, it's much music.
So basically, engineering.我觉得我是要去的，我上个礼拜就在开始。我觉得我这个。
Make them brighter. It's a shame that right in front. What's the remit? I can make it bigger, but it will be horizontal. can make it like this. Okay. Oh, no. Oh, no. Oh. So you turn it this way, it becomes smaller. It becomes 3 times. So no, this is the biggest. I want to sit in front. I want to sit right here. Yeah. And then you see like the surface. So it has not turn on. Wait. Good? Yeah. So grab one. Because I would already know the answer kind of. It's very obvious though. You introduce yourself. I do. So everyone knows.
Everyone, I'm first of all start in 2 minutes so you can try to take your seat. Thank you.
Yeah. Oh, no.
Hi everyone, my name is Turin. I'm from the lab of Dr. Ido Building in the physics department. The most important job of the day is to keep the time. So when I show you my laptop later, please cooperate and wrap it up. And also another announcement is when you want to make questions at the end of the presentation, please wait and I will pass you the microphone so that everyone in the room can listen to your questions. We have a very exciting array of talks in front of us. They really try to push the experimental frontiers all the way from DNA, RNA, proteins to lipids and amino acids. The first speaker, Mei Wang, is my fellow lab mate in the lab of Dr. Ido Building in the physics department. He received his PhD in quantitative and computational bioscience at Baylor College of Medicine, and now he's now doing a postdoc here. In the Roman lab, we have to say that we don't know how to do a lot of stuff, but one of the things we really know how to do is to count. So Mei will now tell us how we count individual mRNA molecules at individual gene loci and the surprising story that he learned from that technique. This is Mei.
Thanks for the introduction. So today I'm going to, so today I'm going to tell you how we measure transcription at a single gene copy and what we can learn about it in terms of the bacterial individuality. And you can see from the background image, here is the individual E. coli cells, and I'm going to tell you the details. So stochastic gene expression gives rise to the population heterogeneity. So the heterogeneity of gene expression has been widely observed in both from E. coli to mammalian cells, both the RNA level and the protein level. And the reason for this is that the gene expression consists of a series of single molecule events that are stochastic in nature. And transcription kinetics is the key to understanding stochastic gene expression because transcription is the first step in the gene expression processes, and the transcription kinetics will drive the protein numbers and fluctuations. And people have been studying the stochastic kinetics of transcription using single-cell RNA measurements for the past almost 20 years. Here I'm just showing you an example where people can follow the production of specific mRNA over time in individual living cells. And based on the trajectories, you can estimate the kinetics of the transcription. And an alternative approach to that is that you can take a snapshot of the cells and measure the distribution of the mRNA number per cell. And this distribution combined with a theoretical model can inform us about the transcription kinetics. And however, these kind of whole cell RNA measurements are very limited in a number of ways. First, it calls grain over contributions from multiple gene copies. As you know, during a normal cell cycle, the gene copy will need to double. And the different gene copies might have different activities. And it also integrates over the whole lifetime of RNA. For example, if you're only measuring the whole cell RNA, you will miss the kinetics of RNA elongation or RNA release or any possible interesting spatial aspect of during the RNA production processes. So our goal is to overcome these limitations by measuring transcription at a single gene copy. And here I'm showing you how we do that, how we detect transcription at a single gene. We start with a paradigm for studying gene regulation. It's a lactose promoter. And we inserted a set of tetal operator array near the promoter in the chromosome. And we're expressing fluorescently fused tetal protein that can bind to this operator array. And in the same cell, we're applying fluorophore labeled DNA oligo that can bind to this mRNA. And here is an example of example image where you can see the lag locus 2 and the different lag Z mRNA signal. And as you can see from this image, the first thing that you might notice is that there are RNA signals that is very close or localized to the to the gene signal. And presumably these mRNA correspond to the mRNA that is being actively transcribed. So in our analysis, what we did first is we measured the distance from every mRNA signal to its nearest gene signal in the same cell. And here is how the distance distribution looks like. You have a very distinct population of mRNA that corresponds to those mRNA that is gene proximal. And here the gray curve shows you a control where we look at the same mRNA but to a different gene locus on the other place on the chromosome. And if we look closer at this population, we look at how the RNA spot intensity changes with in respect to the distance to the gene. And we found that the gene proximal RNA signal is stronger, and that is consistent with the textbook expectation that you have multiple RNA being transcribed at the same time. And under our microscope resolution, we won't be able to tear apart different unfinished RNA. So it will just appear as a single brighter signal. And the second evidence for that is that when we label the 5' end region and the 3' end region of the RNA with different sets of probes. So here, there are two unfinished transcripts. And these unfinished transcripts only have the 5' end region of the gene, not the 3' end region of the gene. So you expect the enrichment of the 5' end region in this population. Well, that is truly the case. And the third evidence we have is when we use a drug to inhibit the transcription initiation. And when you add the drug to inhibit the transcription initiation, after 3 minutes, you already see the disappearance of this population. And 10 minutes is almost all gone. And with this three evidence, we can comfortably say that these gene proximal RNA population corresponds to active transcription. So in our further analysis, we can use a distance threshold to classify the RNA into nascent or mature. And to give you a visual sense, how this is done. So we start from the raw image, and we can classify the gene into active and inactive based on if we have the RNA signal within that distance threshold. And the RNA will be classified into nascent and mature. And now suddenly we have two new measurables that people cannot measure before. One is called nascent mRNA, which is basically the summation of the RNA that is in the proximity of the gene signal. And the second measurable is the fraction of active gene copies. So it's like in this cell, like one gene copy is active, one copy is inactive, so the P-on will just be 50% for this cell. So now we have two new measurables. How to make sure we are measuring things properly, we developed a mathematical model to describe the kinetics of nascent and mature mRNA. So here is how this model looks like. So there is a certain kinetic rate for the transcription initiation to start, and after the transcription initiation, the RNA polymerase will elongate at a constant speed, and the RNA will be transcribed and translated at the same time. And the RNase has a probability, has a kinetic rate of bind to the nascent RNA and start the degradation process. And after elongation is done, the nascent mRNA will become a mature mRNA. So to compare for this model, we can solve the model on a piece of paper and predict the amount of nascent mRNA and mature mRNA in both steady state and non-steady state conditions. And to compare the model prediction with our experimental measurement, we use a turn-on experiment, meaning we turn on the promoter at 0 time point and follow the progression of total nascent and mature mRNA in the cells. And the marker here are the experimental measurements, and the solid curve are fit to the model. And you can see here the data is well described by the model. And there are some interesting features that I want to point out. Like, for example, the mature will only appear after a certain amount of time, because right before this time point, every RNA are still being made on the gene, so there is no mature RNA. And exactly at the same time point, the nascent already reaches a steady state. At the same time point, it reaches equilibrium between new initiation and the release. And by fitting the data to the model, we can extract these kinetic parameters like RNA polymerase elongation speed, degradation rate, and transcription initiation rate. And these values are all consistent with the literature. And so now this is all good, but here we are only testing the model using like a population average. For example, here each individual time point is still an average of a few hundred cells. Can we use a model to predict the distribution of a few hundred cells in each individual time points? And for that, we need a stochastic part of the model. So we add a stochastic component in this model, which say the promoter can have two states, off state and on state, and it can switch between the two states. And the transcription initiation can only happen when the promoter is in the on state. And by adding a stochastic component of the model, we can numerically solve and predict the distribution. And you can see here the distribution of the data is well described by the model. Here I'm only showing you a subset of all these time points, and you can see it's pretty good. And the thing we gain by fitting the data to a stochastic model is like we can have an idea of these stochastic parameters, like how frequent the promoter turns from the off state and how frequent the promoter turns to the on state. That's the same you gain. Okay, so now I showed you like our experimental measurements. We can measure transcription at each individual gene copy, and I showed you our model that agrees with the measurement very well. And if you still remember the title of my talk, we say there's something like a hidden driver of cell individuality. So what I mean is like hidden driver of individuality is like there is some factors that contribute to the heterogeneity of gene expression that are previously unknown. And the first hidden driver I'm going to tell you is like the correlation between individual gene copy in the same cell. So here I'm showing a schematic that tells you two copies of the gene in the same cell can be correlated in time. So to ask the question of correlation, we look at cells with only two gene copies. So here we start with a fast growth condition where the glucose is a carbon source. And here we classify the gene into active and inactive. And when you look at the distribution of number of active copies in these cells, you find that the distribution followed by normal distribution, meaning they are independent. And however, when we change the carbon source to a glycerol, where the cells are growing slowly, you can see there's some correlation if you look here. And the distribution has a very dramatic different shape. Like here, both copies in the same cell tend to be both inactive and active at the same time. And we played with the growth condition. We tried a few conditions, and here's how this correlation changes with the growth rate. And the general rule we found is that when the cells are growing slowly, they tend to be correlated. But when you're growing fast, there's no correlation. And if we see how the correlation varies with different expression levels, we found that in the glucose, there is a slight increase of the correlation, but it's still very uncorrelated. But the glycerol, there's a peak in the median correlation, and then it goes down. We still don't know what gives rise to this kind of, what's the mechanism that gives rise to this correlation, and how this growth rate dependent correlation can have. And the second hidden driver is the coupling of gene expression to the event of gene replication. So we found that there is a pause of transcription events following the gene replication. So let me tell you how we detect the timing of gene replication. So this is based on the phenomenon called sister copy cohesion. That means when the two copies, when the gene replicates, there is a period of time that the two sister copies will be stay together. And on our microscope, we still one single spot, just brighter. So based on that, we can tell where the gene replication happens. And then we look at the nascent RNA per cell versus the cell length. And cell length is a very good approximation for the time in the cell cycle. And you can see it's a follow a simple step shape. And this is almost twofold. That means whenever the gene dosage doubles, there is a doubling of the nascent mRNA in the cell. And this is very too easy to understand. However, when we look at the lactose promoter in a repressed condition, this is not the case anymore. You have a very pulsatile transcription kinetics. Like whenever you have a gene replication, you have a pulse of RNA production. And this is also seen in live cells where we can follow the gene replication in time and RNA production. Here you can see the one copy of the gene become two copies, and then you have the RNA production. And you can analyze many movies of that and analyze the transcription probability with regard to the time after replication. And I think I'm running out of time, so I'll just thank everybody in our Golden Lab, and thanks all the funding sources, and happy to take any questions.
Any question for Mengyu?
I just shout. I don't understand the correlation results. I'm confused. So you're studying the expression of LAC-Z?
Yes.
Controlled by a lac promoter?
Yeah.
In glycerol and glucose?
Yes.
There's no lactose around, right?
No.
Can you interpret this for me in any way? Or I just don't understand why in glycerol, or why at a low growth rate I should see this, and why at high. Do you have a speculation?
Okay, our speculation. Okay, we have like two speculation. First speculation is like there might be some upstream regulator that fluctuates in a frequency that fluctuates in different frequency in the different growth conditions. That only in glycerol only in the slow conditions will lead to the correlation.
But I think perhaps Sepa didn't even understand why you even have the expression of the lactose in that case where there's no lactose. I think perhaps. Okay,
so here we're adding IPTG in both glycerol and in all the conditions.
Oh, it's under an inducible promoter.
Yes.
The promoter is endogenous, but it's artificially inducing. That was not clear.
Thank you. Any other questions?
Following replication, does the RNA symmetrically partition into the two new cells, or is it? You said that there's a transcription first.
Yes.
After replication, do you know where the RNA goes afterwards?
Okay, so we won't really looking into that because you're asking about cell division, right? How the RNA partition into different cells. We won't really looking at that. How partition happens. But it's interesting to look at if it's really like random, if it's followed by binomial partition, for example.
Then let's give me another round of applause. Thank you. Our next speaker, Giuseppe Nicoli, described himself as a computational experimental chemist who received a PhD at University of Geneva in Switzerland and is now studying interaction of small molecules in the cell with a touch cross sheet group, I wouldn't butcher that, in the Department of Biochemistry. Today, Giuseppe will tell us how we can detect lipid orders in the membrane using mechanosensitive probes and molecular dynamic simulations. Whenever he's done setting up this microphone. But let's give him a round of applause.
Okay, I hope you can hear me. Good morning to everyone.
No.
It doesn't work?
I think you just have to project. Can you project it?
Okay. That's fine. I think my voice is loud enough.
So good morning to everyone. Thanks for being here today, and thanks for the organizer as well. So today I would like to give you an overview of our recent work about a mechanosensitive molecular probe that can detect order in lipid membranes. My research focuses on the study of nanodomains in lipid membranes. It's, I think it's fine. You can hear me, right? No. Hello? Okay. So my research focuses on nanodomains in the study of nanodomains in lipid membranes. Lipid phases are very important because they can affect biological activities. But there are only few techniques that can detect lipid phases in a biological environment. Perhaps one of the most used technique is optical microscopy because of its high temporal and spatial resolution. And to use optical microscopy, we need to have a probe. One of the class of compound that is used nowadays is environment-sensitive dyes, and they can change their spectroscopical response according to the environment. And recently there was a new dye that was introduced and commercialized under the name of CPLC. And this compound can distinguish lipid phases in the vesicles. The structure of CPLC is shown here on the left. The chromatin is composed by two butanothiophene petals. I have it in blue. There is a hydrophobic head that can increase the solubility in water. Two hydrophobic petals to have interpolation inside the membranes, and those two methyl groups that can keep the two petals autopolarity in the ground phase. So organic chemists think that this molecule works by twisting these two petals with respect to each other. In particular, they think that in disordered membranes, the radial angle is close to 90 degrees, and in ordered membranes, the radial angle is closer to 0 degrees, so it parallels. And they got this hypothesis from the results in vesicles composed by DPPC and BOPC. For example, if we consider DPPC, we know that at low temperature, DPPC have a phase transition, so it goes towards a more ordered membrane. And CPLC in such a membrane shifts its excitation spectrum towards the red and increases fluorescence intensity. So now the question is this change in the spectroscopic behavior connected to the twisting of the molecule around this radial angle? So to answer this question, we resorted to molecular dynamic simulations, and our aim was also to extract usable information for the design of new, more efficient probes.
Yeah, to take off the lapel one, yeah. Turn that off. This one? Yeah, just use the handheld. Yeah.
So the first step in our project was to get parameters for the probe and also define the parameters. And so we used a quantum mechanical calculation to do that, and we put special efforts on the parameterization and the refinement of beta hydro, which is supposed to be responsible for the mechanical sensitivity we want to improve. And after our refinement, we obtained this distribution of beta hydro from the MD simulation, which actually corresponds very nicely with the minimum observed from the quantum mechanical potential energy surface scan. So this is already an evidence that our refinement was very successful. After the refinement, we started with our simulations, and the first thing that we looked at was the partition of the compound in different membranes. In this video, you can see how the CPLC can partition inside the membrane spontaneously without adding any force. Here the example is with a DPPC membrane. And we did this kind of simulation in different membranes, and we could also estimate the free energy of partitioning using umbrella sampling simulation. And from those simulations, we can extract the free energy of partition and compare with experiments. And our conclusion is very nice with experiments, and this is another evidence that our force field is pretty robust and reliable. So now I'm going to show you some results that we obtained with different membranes of increasing complexity level. So the first example I'd like to show you is in unary phases. We did some simulation in DOPC, which gives orders a disordered membrane at a high and low temperature, as shown by a very high value of lipid and low values for the carbon deuterium order parameters. So from these membranes and this simulation, we extracted the dihedral angle from PPTR as a function of time, and on the right panel you can see the distribution. So the distribution is bimodal, centered at around 70 and 120 degrees, and it's pretty the same at the two temperatures used. However, if we go to DPPC, so at high temperature we also get a disordered membrane with DPPC, but when we lower the temperature to 298, we get a well-ordered membrane, as shown by the decrease of the erythrolipid and by an increase of the carbon deuterium order parameter. If we look at the dihedral angle distribution, we see that there is a very dramatic change in the distribution, which now is single-peaked at around 150 degrees. So there is a change, but now the challenge is to understand if this change is really correlated to the change in the spectroscopic behavior of the molecule. So to do this, we simulated the absorption spectra of PPTR in different environments. To this aim, we used the time-dependent DFT calculation for 250 conformers taken randomly from each simulation. And we calculated the vertical transitions, we convoluted each transition with a Rollinsson function, and we sum up all together all the line shapes to reproduce the final spectrum. So the final spectra are shown here, and they are also compared with the experimental spectra. And you can see that actually we could reproduce the exact shift in the absorption spectra. And this links unambiguously the twisting of this mechanosensitive dihedral to the mechanosensitivity of the probe. So the organic chemist hypothesis was correct. We also wanted to see if the molecule can respond instantaneously to the change of the phase order. So to show this, we devised a simulation where we changed the temperature of the membrane. We started with a PPC delay at low temperature that gives a very well-ordered membrane, and as shown by the erythrolipid. And so the dihedral angle of PPTR is around 150 degrees, as shown before. Then we switched the temperature to 330 Kelvin, and we wanted to melt the membrane. As you can see, the erythrolipid is going up, and the membrane now is disordered. And now the dihedral angle goes down, and now it's fluctuating between 70 and 120 degrees. Then at a certain point, we switched the temperature back to 298 Kelvin, a certain point here. And now the erythrolipid goes down because the membrane starts to order again. And at the same time, the dihedral angle starts to go up again 250 degrees to the original values. So it means that the molecule can really follow and respond instantaneously to the change of the phase order in the membrane. So now one step forward to a more complex membrane, we used the sphingomyelin cholesterol mixture with this ratio to simulate liquid ordered phases. So this mixture gives always ordered phases at two temperatures, as shown by high values for the carbon deuterium order parameters. So from this data here, you can see that the membrane seems to be pretty the same at these two temperatures. So one could say, okay, maybe the lateral forces in the membrane are similar. But if we observe the dihedral distribution in PPTR, we can see that there is a pretty big difference between the distribution. And if we observe the shift again in the absorption spectra, we could again reproduce the exact shift, and we can see that the absorption spectra are different in these two membranes. So there are differences in the lateral pressure profile. And now the last step was to use even more complicated membrane. So we wanted to see if PPTR was operative also in a phase-separated membranes. We try to emulate the situation shown here in the pictures where we have the separation of the phases. To do that, we prepared this big membrane where we have nanodomains composed of sphingomyelin cholesterol embedded inside a bigger DOPC matrix. And we added inside and outside the membrane PPTR colored in yellow here. So we simulated for more than 1 microsecond this simulation, and after the simulation time, you can see that there are a few molecules that diffuses away from the inner nanodomain. But still, you can observe that there is this circular wrap-like domain inside. And if we analyze now the distribution of the PPTR diagonal angle, you can see that inside and outside they show a different distribution. So we can distinguish the lateral forces even in such a big and complex membrane. So to conclude, we developed a robust force field for PPTR. We observed microscopically the sensitivity of this compound in different membranes, and we explained unambiguously its working mechanism, and we could observe that the probe senses order delays to different composition and responds instantaneously to limit for the changes. And now I would like just to thank my advisor, Emma Teshkochi, the experimental collaborators from Geneva who synthesized the molecule and also provided the experimental results, the funding, and all the CBQB group. And thank you for the presentation.
Thank you. Any questions?
Do you measure the pressure profile of the membrane?
He asked if I measure the pressure profile of the membrane. I didn't yet, as on the plan, but I did some experiments where actually we play around with the pressure coupling with Barostat, so to try to change the surface tension of the membrane. We could also observe that the molecule is sensitive to the surface tension.
So is there in what other way you measure the mechanosensitivity of this probe?
You mean
what are the parameters you say that okay it shows this kind of characteristic, it is a mechanosensitive of it?
Well, we looked at the twisting because this is the one which, the twisting angle between the two pebbles is the one that people believe was responsible for the mechanosensitivity. So we looked at that, and that's the other one that we looked at.
Good question. Does it always twist one way, or it's stochastic?
No, it can twist both ways. The molecule has a plane of symmetry. So actually, the diagonal angle that I show you is within 0 and 180 degrees. So every time it flips on the other side, I put it back always on between 0 and 180 degrees because we have a plane of symmetry.
And thank you. Thank you. Next up we have John Smith. He's an expert of making movies for very, very small. John received a Bachelor of Science in Material Science and Engineering here at UIUC, and he loves the weather here so much that he decided to stay to pursue a PhD here in the same field in the Chen Group in the College of Engineering. Today, John will show us how he and his colleagues look at single particle dynamics using liquid phase electron microscopy, or rather what he calls, and also my favorite way for him to call it, electron cinematography. John.
Don't use the microphone. Just turn that off. Only for questions.
Oh, people can really hear it.
He just needs to project and use the.
Let us know you can hear it.
Am I on board? No. You mind? Is it on? Hit it. Just hit the mic.
Okay, how about now?
Yeah.
Okay. Yeah, so thank you for allowing me to present. I'm Johnny from the Material Science College.
Can everyone hear?
No.
Okay, can you try to a little louder?
Or you can just hold the microphone instead of.
Or put it on your other side because you will be looking.
Yeah. How about now? No.
Can't hear you.
How about now?
Yes.
Okay. Yeah, so in our group, what we're trying to do is develop
Use the hand.
Just hold it. Hold it. Okay. Well, we're getting a lot of feedback from the handheld mic.
Can you use this one instead?
You can. I mean, well, let's see what the feedback.
All right, is it okay now?
Okay. So which one's the other one? Yeah. But is the other one still on? Turn it on.
Okay, so here we go. Yeah, so in order, we're working on trying to develop new kinds of imaging techniques and also analysis techniques to understand phenomena related to cell physics or chemistry or biology on the nanoscale where there's trying to meet each other, which is the nanoscale. So what do aquariums have to do with it? So if we start on this scale, on vacation, I'm sort of this person right here who might go to an aquarium to have a relaxing time, observe these beautiful sights. But in the lab, I'm more like this scuba diver down here who might use an aquarium to learn how fish use their mouths to eat or their fins to swim just by watching them in action. And you do that in these various sorts of complex environments because he sees them with his eyes. So I won't really talk about fish today, but the game is the same. So proteins accomplish all kinds of amazing tasks, not just because of the complex structure, but because of the complex dynamics of it. And so one of our big goals is to really see these processes in action. So how can we do that? As many of you know, in order to design to see something, there are sort of fundamental limits. But what we can do instead is use electrons, which have a much smaller wavelength. And if you look at something like a quantum dot, instead of the spot, you might see the fluorescence. With electrons, you can sort of see the actual atoms inside that quantum dot. But for proteins, it's not quite so simple. One reason is that proteins have very little contrast in electron imaging. So for a long time, the sort of classical way to see proteins in an electron microscope was to sort of surround them in this very dark sort of metal stain. So here I'm showing some examples. We can see A1 fibers, or ferritin, or even something so fine as this DNA strand where we can trace the contour. But it wasn't really until techniques like chromatin where people learned how to make even out of many thousands or even millions of very noisy images and put them together into this beautiful high-resolution structure of a protein. But of course, you're wondering what about motion? So this is where our other type of aquarium comes in. So in our group, we're working on a technique called liquid-phase transmission electron microscopy. And what we do is to look at proteins or other systems in the vacuum environment of an electron microscope. We trap them in a tiny sort of aquarium or capsule. So around 5 years ago, people devised this chamber made out of silicon nitride. But for me to see proteins, we have one more trick, which is making these chambers out of graphene. So graphene is this sort of atomic thin membrane of hexagonally bonded carbon. And so it gives us actually a wide variety of benefits. So because it's so thin, we get the most bang for our buck in terms of contrast. And at the same time, that lets us use low doses of electrons so we don't destroy our sample when we image it. And because graphene is also very thermally and conductive and also very strong, it sort of protects our sample as we image it. So the system I'm going to talk about today is a structure called nanodiscs. So what that is, it's a tiny piece of the bilayer. It's surrounded in a helical scaffold protein, two of them actually, and then inside you can put another you're interested in studying, and because of our all related and to fundamental biological processes, that's sort of why we into the structure to study living proteins. And the utility of this kind of structure is that it allows you to look at membrane proteins without breaking the whole cell membrane along with you. And so putting all these things together, instead of these sort of solid-embedded structures, we can get images of nanodiscs that are actually in a liquid environment, and not just images, but actually movies where they start to wiggle around. So this was pretty exciting to us to achieve this. And so today I'll try to talk about how we're trying to learn from these real-time images. So first we started kind of simple, just looking at sort of the outline of these projected shapes. So even from that, we can get simple parameters like a diffusion coefficient or a rotational diffusion coefficient. But we took this opportunity to do sort of a sanity check to make sure things are working properly. So what I mean is, first we sort of treat the projections in an ellipse. So that projection might vary because the net disks are rotating or because their shape is actually fluctuating. So the liquid memory is fluid-like. And then what we did is actually look at several different types of net disks that have different dimensions and different properties, and try to see like does what we're seeing really make sense based on our expectations. So the first thing we did is we compared both small diameter net disks and large diameter net disks. So the way we do that is by changing the size of the belt they use to make them. So a net disk with a bigger belt will eat more for a little bit of day. So yeah, so it's a sort of a simple verification, but we can sort of match what we did in the experiment by changing this belt size based on the dimensions we expect. So this is sort of a static verification, but we also try to take the number of and see what changes, what happens. And so as you might expect, the analytics are a little bit floppier. So we look at their sort of dimensions over time, there's sort of wide deviation. And so to sort of look back at the sort of dynamic aspects, we turn to dynamics simulation. In this case, coarse grained dynamics simulation. So we put these models, ran some simulations, and again, tying back to our experiments, we can compare those results from sort of simple particle tracking to the results from the simulation. So there's a little bit of mismatch because the time scale is very different, but at least very qualitatively, we can see similar phenomena. But there's actually quite a lot more information you can get from these types of fluctuations. So those of you familiar with sort of giant vesicle research may know that by watching the way that the denominator flops around, you can actually get some physical insight. So those fluctuations are related to how stiff the membrane is, how bendable it is, and so these are things that we can also extract from these types of real-time information. But yeah, and so this is another thing where we can sort of use the molecular simulation to kind of calibrate ourselves. But a little bit more surprising thing is, unlike in these kind of simulations, in some cases, when we look at disks for a long time, we actually observe that they really have the ability to adopt like very large scale fluctuations. So as you watch here, you'll see like the disk settles around, and you can see sort of budding or finger-like projections, and then comes back on itself. And so in that process, my mathematics initially a sort of core domain, and then over time, those sort of are partitioned into subdomains that eventually come back to the core. And then one thing we try to think about is what types of sort of mass flow or diffusion are involved in those processes. So these arrows are sort of pointing where the liquid is moving between frames. And this is sort of consistent. We do this fairly consistently. But another sort of surprise is when we looked at these methods that don't have membrane proteins inside them, what happens when they undergo this kind of budding phenomenon is they will actually eventually become destabilized and sort of totally develop. So our idea is that somehow the presence of this membrane protein inside the nerves helps these structures to recover from these very drastic fluctuations. And so, I guess going further, one thing we're trying to do is treat them as more than just blobs in the shape, and really think about what's going on inside the membranes. So what we can do that is by sort of predicting the type of image we would expect based on the negative protein in solution, or based on the membrane protein diffusion inside the membrane. And so here is an actual snapshot from one of the simulations. And what we're trying to do is build a sort of workflow where we can compare external images to real sort of molecular level coordinates inside these structures. So with that, I will wrap up. So we're trying to, essentially we're trying to come up with new ways to think about structures at the nanoscale in real time. So there's simple but also some more surprising results. And in the long term, what we want to do is not just these fluctuations, but also look at dynamics that are really related to function. So this is just some ongoing work with Paul Simon's group. And with that, I should thank our collaborators and the Department of Biochemistry for helping prepare this. This is my group, and we talk a lot about USTM and partners in my place. Happy to take questions.
Yes. What is the spatial and temporal resolution of this technique?
So in principle, the spatial resolution. Okay, the question is, what is the spatial and temporal resolution? Spatially, in principle, we have the resolution of electron microscopy, which is sort of ancient resolution. But the sort of signal to noise ratio makes it a little bit less. So in practice, for these movies, the resolution is around 22 nanometers. Temporal resolution, in this case, so the frame rate is a few hundred milliseconds, but that's something we can push much lower. It's just sort of a trade-off between contrast and
it was unclear to me with the movie where you were seeing supposedly the asymmetry. There we go, yeah. It was unclear, are these actual pictures or are they simulations?
So these images are fake. This image is real. So
wait, which is real?
So this one here is real. The
Which?
one on the right, the top right.
Yeah, okay.
So
that's real.
Yeah. So I guess what we're trying to look at how contrast variation inside the boundary is related to where, for example, where the neuron is inside that boundary. But we do that by sort of picking different types of possible energies and then trying to match that back.
Do you have to do anything to graphene so it doesn't stick to small discs?
Yeah, that's a good question. So yeah, the question is, what do we do to the graphene to prevent sticking? So yeah, in some cases, it's not necessarily a bad thing for us, because if you imagine that this matter is just based on its size, so it's on its size.
So all of that it's stuck.
Yeah, most likely it's stuck, but it helps us, like, if it was using a Stochastic current, it would be very difficult to see. So sticking can give us a little bit of help in terms of keeping it where we want it to be. Yeah, but there are ways to sort of functionalize the graphene, so it's not like this is chemically attached.
Yes, just one more question.
So what is the typical thickness of the liquidity of cells? Like,
so it depends on which type of liquid cell we're using. So for the silicon nitride cell, that can be, let's see, maybe hundreds of nanometers, maybe 1 to 200 nanometers. Yeah. But for the graphene liquid cell, I would say tends to a few hundred nanometers. Tends to a few hundred nanometers.
And thank you for that talk and a very exciting Q&A. Here.
And this is our last talk session. Our last talk concerns a very exciting problem in biology, how to sequence for proteins. Because it's not as approachable, as accessible as sequencing for DNA and RNA. Our next speaker, Kumar Sarthak, from the Ashimantiev group in the physics department, has a way to cheat the system and sequence for the single amino acids in the proteins. Kumar received his Bachelor of Science in Indian Institute of Science, and he's now pursuing his PhD here. Once again, this is Kumar. Okay, let us know you can just project.
Can you all hear me? Right, so I'm Sarthak from the Experimental Lab, and today I'm going to talk about recognizing 20 proteinogenic amino acids using an error lysine nanopore. So proteins are ubiquitous in a healthy human body, but any kind of error can lead to a single point mutation, which could lead to a variety of diseases like dementia, ALS, and even breast cancer, which affects nearly 250,000 people in the United States every year, out of which 50,000 are fatal. So it's very important to know what the exact sequence of the diseased protein is, so that we can go back and see what was the molecular origin of that disease. And it's also very essential that the technique that you would probably use for this should be single molecule because of the low abundance of these proteins at the early onset of diseases. Now, we can do that by looking back at the DNA sequence which led to the protein sequence. Now, DNA sequencing is a solved problem. There are kind of three major companies that do it: Illumina, PacBio, and Oxford Nanopore Technologies. Now, what I would like to draw your attention to is simple but very effective technology of nanopore sequencing. Now, typically what you would have is you would have a nanopore that is embedded in a lipid bilayer, and you solve it in an electrolyte, let's say potassium chloride, and you apply an electric field. Now, what happens under the electric field is ions start flowing, your positive charge is going up, negative charge is going down. Now, you can also add an analyte to the system, which in our case is DNA. Now, once the DNA is passing through the pore, each nucleotide will block the flow of ions to a different rate. Now, looking at the residual current, you can go back and see what the DNA sequence was. Now, can you use the process of DNA sequencing to sequence those mutations that I talked about before? And yes, for this ALS mutation, you can sequence it and diagnose it. But for this cancer mutation, it cannot be diagnosed because it's a mutation that is induced as a post-translational modification. And here is where comes the importance to know the difference between the genotype and the phenotype. So it's very important to go back and sequence and have a de novo protein sequencing. Now, even though we have known proteins since a very long time, there have been only two kinds of techniques that are widely used for protein sequencing, and that's because there hasn't been any kind of evolved biological machinery that can read or even copy protein sequences. Now, the problems with these methods, Edmond degradation and mass spectrometry, are they're not single molecule, and they require Edmond degradation requires less than 50 amino acid proteins, and mass spectrometry has a very low detection limit, and also requires a large amount of sample. Now, can we use the same technique as we use with DNA? Can we do nanopore sequencing? Now, we can look at an ideal case where you can have a peptide that goes through a nanopore. You apply an electric field, it goes through it, and you look at the current, and it just gives you the peptide sequence. Now, it's not as simple or as easy as for DNA because of a few differences. Now, one major difference is that we have 20 amino acids as compared to just 4 DNA nucleotides. And the resulting current would be from all the 20 different amino acids, and it would be very hard to distinguish between them. And another major difference between DNA and protein is that DNA has a phosphate backbone, which is negatively charged. So an electric field pointing upwards can easily capture your DNA. But that's not true with proteins, which can have varied charge. Now, what if our protein was homogeneously charged? Now, this is what our experimental collaborators had in mind at the University of Cergy-Pontoise, France, where they designed a sequence of just arginines, which is positively charged. And at the end of the sequence, you can add another amino acid, which can be one of these 20 different amino acids. So you can use this positively charged region to capture the peptide, but also have an amino acid which could be any of those 20 different amino acids. Now, what they did was they applied an electric field pointing downwards and let it flow through a wild-type aerolysine nanopore. Now, typical experimental data looks something like this, where you have an open pore current, and once the peptide goes inside the pore, the current decreases, and this is what you observe for a blocked pore. Now, you can do this for, say, a single amino acid, let's say it's serine, and what you would observe is a blockade current that goes as a Gaussian distribution, centered around a mean, and has a standard deviation. Now, let's say we do it for a mixture. Now, you can do it for a mixture of electrically charged side chains, and what we observe is clear distinguishability between these 5 amino acids, even in the mixture. Now, can you go ahead and do it for very structurally similar amino acids? Let's say leucine and isoleucine, which are structural isomers, which have a molecular weight difference of 0, and you can still see that you can distinguish between a peak of leucine and a peak of isoleucine. Now you can just take a leap of faith and go for all 20 amino acids and try to compare the mean blockade values. And what we see is very interesting. Using a wild-type aerolysine nanopore, you can sequence 13 out of the 20 different amino acids. Now the regions that are not that great are this region and this region where the blockade values are similar. Now we need to go forward and use the sequencing device, for which we need to improve its resolution. To improve the resolution, we need to know what is the molecular mechanism that is kind of leading to this exquisite distinguishability shown by this aerolysine nanopore. How does it work? So to do that, we would go to a computational microscopy, and which I think is the all-atom molecular dynamics method. So what we do is we take the protein structure from the protein data bank, the structure of aerolysine, embed it in a lipid bilayer, and solvate the system in water, and add ions corresponding to 1 molar KCl solution. Now you can evolve this system in time using the molecular dynamics method. Now what you have in an MD simulation is the coordinate of each atom in the system at all points in time. And using that, you can calculate the electrostatic potential at -100 millivolt, which is what we had before. Now, what the peptide would feel as it enters the pore is something that's at the central axis of the pore. So we can look at what the electrostatic potential profile is at the central axis. Now, if you look very closely, you'll see something very interesting. At this region, we see a constant potential region, and which is flanked by 2 potential barriers. Now, we felt that this could possibly be a trap for the peptide in this process. Now, keep this in the back of your mind as we go forward and actually simulate a peptide translocation through an nanopore. Now, peptide translocation occurs at a typical time scales of 10 milliseconds. Now, that is not something that you can do using molecular dynamics. So what we do is we attach a fictitious spring to the peptide and pull it at a constant velocity of 1 angstrom per nanosecond. And and this is how it looks. Now you can also calculate what the force that you are applying to pull it at a constant velocity, and it looks something like this. You have 2 constrictions, one at the top and one at the bottom over here. Okay, now that we can simulate translocation of the peptide, we need to go and calculate what the anic current through the pore is. Now, the anic current distinguishably that they had seen in experiment was of the order of 0.05 picoamps. And to get enough number of ions flowing so that you have a less error lower than this would require a simulation time of nearly 700 microseconds, which is also pushing the limits. So what we do is we use a recently developed continuum model for nanopore anic current calculation, which is very fast because it's a continuum model, and also it is atomically precise. The current values that we observe over here exactly match what you will observe if you do a complete MD simulation. Okay, now that we have a method for calculating ionic current, we can go back and look at what the ionic current to the pore was as the peptide was translocating, and I have put, and this is an 8-arginine sequence, and I have put the experimental value for the 8-arginine sequence. Now, as the peptide passes through the pore, you can see that initially it starts closer to the open pore current level value. It goes down nearly to 0 at the first constriction. Again, it goes down nearly 0 at the second constriction, and then it goes back up as the peptide exits the pore. Now, what's very interesting is what happens in this region, where the experimental average matches very well with what we observe using SEM. Now, this is very interesting. Now, what we think is this is a region where there is the sensing that's happening. A peptide enters this region, and there the experimental value and the MD values match. Now, how can we confirm this? So what we do is we select frames from our assembly trajectory and computationally mutate the last amino acid in our simulation. And what we observe is very fantastic. There is clear distinguishability between those 5 different amino acids in this sensing region. And you can see it matches really well with the experiment. Now, what we think is the molecular mechanism that is going on is that once a peptide enters the pore, it can kind of diffuse around in this constant potential region, and that what we call as a single molecule trap. And once it enters this region, you can clearly distinguish between the different amino acids based on the ionic current level. Now that we know what the molecular mechanism behind all this is, can we go forward and now improve the resolution of our sequencing device? Now, one way to do that would be to check what's the difference between leucine and isoleucine. This is our test case, molecular weight difference of 0. They're very close to each other, and we have less than 70% identification in the experiment. Now, you can use the same data and put an idealized Gaussian with the mean and standard deviation as we had observed. And theoretically, you can increase the residence time and see that at a residence time of 200 milliseconds as compared to 17.5 milliseconds in our experiment, you can have a greater than 99% probability of distinguishing leucine and isoleucine. Now, this is very interesting. Can we now increase the residence time in an experiment and see this distinguishability? Now, this is what our experimental collaborators did, is that they further reduced the potential from minus 50 millivolts to minus 40 millivolts, and they could see a distinguishability greater than 92% for leucine and greater than 99% for isoleucine. Now, this was also a low noise setup. Now, you can go forward and also change the electrolyte to be a 1:1 ratio of KCl and KNO3, which increases the residence time even further, and you have a greater than 95% distinguishability for leucine. Now, looking back at our data, now can we also change the blockade current levels of these of the regions that are difficult to distinguish? Can we move them out somehow? Now, we use this chemical modification that is very common in nature. It happens as a post-transcriptional modification that you can add a nitrate to your tyrosine molecule. Now, can you add this nitrate and try to move it? And what we observe is once you add a nitrate molecule to your tyrosine, the blockade current moves out of this difficult region. Now, you can also do this for a methionine amino acid and using a sulfoxide tag, and you can see that now it also has moved out of this difficult region. Now, using all of that, what we can sequence is now 17 out of the 20 amino acids using just the wild type hydrolysis nanopore. And this truly, I believe, is the first step towards single molecule protein sequencing. So the work that I described has recently been accepted in Nature Biotechnology. And I'd like to thank the members at Experimental Lab, Professor Experimental, and our collaborators at France and Germany, and the funding agencies. I'm happy to take questions.
Thank you. More questions? Yes, a lot of questions.
Would the neighboring amino acid have an effect on the sequencing? If you're like doing a full-length protein, so you have like different sequences of amino acids, or the nearby amino acids affect the actual potential that you.
So our method is something different to what you are expecting. So in DNA sequencing, where you would have is like an entire chain that you can put through and you sequence it one by one. Now, in our method, what we need to have is ligate the amino acid before you go into the sequencing process. So you chop off the terminal amino acid, attach it to your carrier sequence of arginines, and then you allow it to pass through the nanopore. So it's not exactly same as strand sequencing as what you would describe.
Did you use an HPLC for that?
The data, those are nanopore data. It's you mean before adding the sample? I don't think so. The samples were pure. You're asking HPLC to kind of purify the sample.
Yeah, so if your peptide has some kind of secondary structure with the scenario network, or the nanopore site is such that while the peptide passes the nanopore has to
So what we have here is an 8 amino acid sequence. So we don't expect any kind of secondary structures to have to be there for us. Yeah, so that's where his question kind of comes out for bigger proteins. What we think of is having a method to chop off the last amino acid and attach it to a carrier sequence, a 7-arginine sequence, and then allow it to pass. So once it is passing through the nanopore, there is no, it's not the entire protein that is passing. Yes.
Is there any other way we can differentiate the similar amino acid other than attaching RNA? Like, have you thought about?
Like, you mean other than nanopore sequencing?
No, like attaching like NO2.
Oh yeah. So that's something that we are moving forward to, like have specific amino acid specific reactions, so that you can kind of move those difficult amino acids over here out from that area and have a blockade current that is completely different from what you have for the neighboring acids. That's what you're saying. Like, so there could be other chemical reactions that do this. Nitration is something that we observe in post-translational modifications. That's what we tried, but I haven't looked at other chemical reactions.
What I mean is, other than the chemical reaction, like by intrinsic itself, like, is there any other we can think about, like?
So one way we have thought is of engineering the nanopore itself to have a higher barrier of the single molecule trap, so that you can reduce this standard deviation even further and or distinguish without needing any kind of chemical reactions. That is one way that I have thought of, but there could be other ways that could happen.
And let's give another round of applause for Kuma. Before we go on to our next 15-minute break, I would like to draw your attention to the back of the room where there will be voting forms. Please don't vote yet because there's one more session ahead of us, but if everyone can take one voting form, that would be greatly appreciated. And now we're going to have like 10 to 15 minutes of break. Thank you.
You just made it. Was the just the volume up too high?
If the speaker speaks too low, I can drop it.
I guess
I still don't know.
Does it seem like you have people? I mean,
Probably need to hold the I'll go to the back. Have some
So you're gonna try to test it out, but that last one got it's fine. Whatever. Yeah. Those on the YouTube, they serve it different. And then I don't I think yeah, they see for and then how mic was working just fine.
it was okay. Good.
Yeah.
So I think possibly that it was just basic moments.
And that's also an issue. Very low. The chemical mics, there was a feedback on that. So it's this magic thing where it's like, okay, it was a problem before, now it's not a problem. So it's this weird interplay between the volume of the speaker and
I wasn't thinking about this.
yeah. So we sort of sit down and write down some notes for next year. But I think it's much worthwhile to do you know
I don't know. They have something called ITS. Maybe we should just talk about this one has ITS.
No, I mean, that's part.
How is that working? You're definitely.
I have another laser.
No, it's not. Yeah.
Put that on all.
They want me to indicate my keyboard, but I don't have like press the key immediately, like side of the keyboard that can help me identify. I don't have that.
I don't think so. It was your source. I don't know, a little bit of time I posted. So you have yeah, okay. Just wanted to talk. So I talked to Alex. Definitely.
Yeah, I have a different type of one. Make a difference. Yeah. Okay, sorry.
Yeah, so I believe there is. There actually isn't too much. Yeah, I think microbiology is
No, I'm using my
One of the chemistry departments. Just to give you a sense of time.
So, there's other groups that try to use droplets.
I'll be here. No, I'll be here.
Today has nothing to do with I've tried tropic. It does work, but I'm not the droplets.
Wait, but is it presenting its own?
So, like the talk I gave.
It's doing its own job.
Oh, like it keeps going.
Yeah.
It might be, yeah, it might be kind of laggy or something like this.
That's fine, I'll try.
You can take almost droplets and label them as different colored RNAs. You can measure
I think you get photo bleaching of the
Make this paper.
that's like you're actually
So our particular assay.
I can go like slightly further, right? Like
But if you're doing microbiology, usually you can drive up the droplet size, but decrease the concentration. We've been trying to avoid that. I mean, for these people, artificial yeah, so problems like
is that my water?
All over the
also, like, I'm not, I was told by an manufacturer. And then the second problem I run into is seems like the droplet solidify buffer. This is right. It seems like they solidify the buffer that has already.
Well, it's also
the trapping might cause I don't know for sure. This I've done a lot of my research specifically on this
मुझे भी ऐसा लगा था लेकिन कुछ बन नहीं था कंफ्यूज हो गया था So the main questions you're asking is the question of
It's kind of like these guys right now. They're having the first paper that I've seen but I also thought that
the scientific question.
I like your sweater.
Droplets, and also is there a bias toward getting our current droplets because we form our droplets.
Can I do a soundtrack? Hi, can you hear me? Hello, hello, hello.
You can use W action. Are you sure you get the viscosity if you have something like
Adjust the gain.
I saw their poster.
There's two separate ones, so
Do you need someone else?
I can alter it up real quick, because a lot of times it's okay. You gotta make sure it's both kind of
And we're trying to do less guardian cytosis. Where this is work like you got that.
Transporter है
turn up a little more than
Oh, yeah. I might go to that. It's better
Okay, we're going to start the third session, the final session of today. Please that you sit down. So the first speaker is Yuhan Wang from Martin Grubel's group, and today she will tell you about us about how cell volume controls protein stability and compactness on the unfolded state. Thank you.
Thank you. Good morning, everyone. I'm Yuhan. I'm a third-year graduate student from Microbial lab.
Today I'm going to talk about cell volume controls protein stability and the compactness of the unfolded state. The interior of the cell is dynamic. Water molecules flow in or out the cell membrane, changing its volume. Here is a video showed two cells changing their volume as in a cell routine cell cycle or as a response to the extra environment change. Protein perform their functions mostly inside the cell. The proteins, I don't know why that happened. The protein's function is closely related to their structures. If a protein wants to interact with any other molecules, they're required to change their conformation. This conformational change can be really tiny, as only a few unbinding of the hydrogen bond, or this conformational change can also be really large as the protein is unfolding its entire thing. So the question we're trying to figure out here is we're trying to figure out the relationship between the cell volume change and the protein inside the cell. So when we change the cell volume, a lot of things is changing inside the cell. For example, ionic concentration is changing, micromolecule crowding is changing. We want to figure out whether those changes will affect the protein behavior inside the cell. Especially for protein that does not have a well-defined tertiary structure, we want to know whether those proteins will be more sensitive to the cell volume change. To answer those questions, first, several in vitro studies have been done. So we're going to use the melting temperature here to indicate the protein stability. The melting temperature means at this temperature, the protein is equally distributed at the folded state and the unfolded state. So if we take a look at the ionic concentration in vitro, for certain types of ion within a certain range, the in vitro study showed the increasing of the ion concentration is not changing the protein stability that much, as shown here. However, when it comes to micromolecule crowding, which is also a very important factor inside for the protein stability, we can see in vitro data shows as we increase the micromolecule crowding, the stability of the protein is increased as the melting temperature is shifting towards higher. As we decreasing the micromolecule crowding, the protein stability is decreased as the melting temperature is shifted towards lower. So the micromolecule crowding is actually affect the protein stability. That's the result from in vitro experiment. When it comes to in cell, will that be the same? Well, we don't know yet before we do any experiment. So if we change our cell volume, let's make a guess. We will going to change the in-cell crowding as well as the ionic concentration. So probably those change will not only affect the stability of the protein, but also change the compactness of the protein. So to study how different ensembles of protein are affected by cell volume change, we first need to figure out three things: how to modulate free cell volume, how to monitor protein stability as protein state, and how to modulate protein partition. First, how to modulate cell-free volume. We're going to do that by using osmotic pressure. So we all know if we have a cell, we expose it in a hyposmotic media, the cell will increase its volume. If it's at a hyposmotic media, it will decrease its volume. And this relationship can be quantified if we do a 3D confocal imaging. As we image the cell, we reconstruct its surface and then calculate the volume inside the cell. And this can be plotted as a quantitative relationship versus the osmotic pressure change with the change of the volume. As the osmotic pressure change is negative, the volume is increasing. When the osmotic pressure is positive, the volume is decreasing. So, in our experiment, this changing can go up to 30% in either direction. Here is a video showed a cell shrink its volume. So we know as the cell is exposed in the hypersmotic media, it's decreasing its volume. Basically, it's vaginate its membrane. So basically, if we have a cell, we have a thinner cell that will look darker. So why is that happening? So those black spots appear over here are just the shrinking of the cell membrane. Oh, dramatic. All right, now we find a way to modulate cell-free volume. Let's think about second question, how to monitor protein states. We're going to do that by using FRET. If we have a protein, we attach our protein with a green fluorophore and a red fluorophore. When the protein is folded and those two fluorophores are really close to each other, if we excite the green, which is donor, the donor will transfer energy to the red acceptor. So after excitation, we will get a red signal at around 600 nanometers. However, if the protein is unfolded, the donor will be far away from the acceptor, then there is no energy transfer between the donor to acceptor. So if we excite the donor, we will get just a green signal at around 500 nanometers. So by looking at the signal, we can tell whether the protein is at unfolded state or folded state. For folded protein, we'll get more red, less green, and a low donor over acceptor ratio. For an unfolded protein, we'll get more green, less red, and a high donor acceptor ratio. All right, now we find a way to monitor protein state. Let's think about how to modulate protein partition. The model protein we use here is called PGK, phosphoroglycerate kinase. It's an enzyme used in glycolysis. So we made 3 mutations for this PGK to make it less stable. And the melting temperature of this mutant will be 35 degrees. We choose 35 degrees because this temperature is higher than room temperature, so this protein will remain stable at room temperature. Also, 35 is not so high, so it won't kill the cell. All right, so when the protein is at room temperature, it's mostly folded. If we heat up the cell to increase the temperature, the protein will become more and more unfolded. The population of the protein being unfolded will increasing. So by changing the cell temperature, we can change the population distribution of the protein. So which will be our sweet temperature to do our experiment? If we do our experiment at room temperature, then most protein will be folded. That's good, but our volume perturbation might not be large enough to drive the protein to the unfolded state or extend the protein. So our sweet temperature will be at 35 degrees, which is the melting temperature. This temperature is in the middle of the transition, so even a tiny little bit of the volume change can drive the protein to the unfolded state. All right, now we answered all of 3 questions. Let's do our experiment. This is our experiment setting, and we can divide this setting into 2 systems. The first left-hand side system is called volume control system. The right one is the imaging system. So our cell will expressing a FRET labeled PGK, and it's performed here. And then we illuminate the cell by blue LED. Because of FRET, we will get both red signal and a green signal. And to modulate the cell volume, we will need this system. So we'll have 2 bottles that contains 1 isosmotic media and 1 non-isosmotic media. The bottle will sit in the water bath that can control the temperature, and we will have a distributor that can rapidly switch between those 2 bottles. So the media flows through the cell will be rapidly changed as we change the bottle. So the cell will shrink or expand according to different osmosis. This is an example of the cell goes through the volume change cycle. As we can see here, the cell is exposed in hyposmotic media, so it decreases volume by around 30%. At the same time, we can see an increase of the red signal and a decrease of the green signal. Of course, an overall decrease of the D/A ratio. This means when the cell's volume is decreasing, the protein is being folded, or the protein is being compact by the cell volume. And then as we recover the cell volume, the signal will recover as well. All right, that's just one single example under hyposmotic media at room temperature. If we do the same thing multiple times for different osmosis and different temperature, we'll get a full image like this. If you take a look, this is a control. So at room temperature, when the cell is always in isosmotic media, no volume change, then the signal will be the same. But if we increase the cell volume by around 30%, then we will see a slightly increase of the green signal. If we decrease the cell volume by 30%, we'll see a slightly increase of the red signal. Those signal is very tiny, not very significant. That's because, as I said, at this temperature, the protein is mostly folded, so the volume modulation cannot drive it all the way to the unfolded state. However, if we take a look at the 39 degree, which is already in the middle of the transition, then as we increase the cell volume, we can see a dramatic increase of the green signal. As we decrease the cell volume, we can see a dramatic increase of the red signal. That indicates the protein is being unfolded or being expanded when the cell increases volume, and being folds or being compact as the cell decreases volume. The same thing happens in the middle temperature. So overall, if we compare this row to this row, at this row, which is proteins mostly folded, they are more sensitive to the cell volume change. All right, so how to explain our data? We have two scenarios to explain this. First scenario is called melting temperature shift, which is the same idea as the in vitro experiment, as the protein stability is being affected by the crowding change. As we increase or decrease the crowding, the protein structure is not changing, but the population of the protein is being redistributed. So the melting temperature shifts towards higher or lower for about 2 degrees. So the significant feature of this scenario is that if you take a look at here, compare the crowding increasing to the basal, there is no DOVA difference. So it's basically 0. And compared to here, really high temperature, it's also no difference. So it's also 0. And we can find a peak at the melting temperature. The second scenario is called changing in unfolding compactness, which is the protein stability is not changing. So the melting temperature is staying constant, but as the crowding increasing or decreasing, the protein structure is being changing by either compact or expand. So the D/A signal is changing. And the significant feature of this scenario is as the temperature is really low, there is no changing, but as temperature is going up, there is change, and the change does not have a peak at melting temperature. So, which scenario will explain our data better? If we plot our data as those dots on the scenario curve, we will see for the crowding increasing condition, both of them can kind of explain our data, because we cannot really tell if there is a peak happen in between or not. But when the crowding is decreasing, the scenario 2 explains our data better, because obviously there is no peak happening in between. So this indicates the the changing of the cell volume not only changes the stability of the protein, but also change the structure of the protein by changing its compactness. Especially for those proteins that does not have a well-defined tertiary structure, they are more sensitive to the cell volume change.
Sorry, you need to.
Okay. So for conclusion, we find a way to modulate cell-free volume by perturbing osmotic pressure. Cell volume change not only change the protein stability, but also affect the compactness of the protein, and unfolded protein is more sensitive to this change. I would like to thank my advisor, Martin Rubla, and my mentor, Shahar Caitlin, and all other lab members. I would also like to thank NSF for funding and the CPLC for giving me the chance for presenting here. And thank you all for listening. I would like to take questions.
So in the interest of keeping on time, we actually don't have time for any questions.
All right.
So just ask her after the symposium during lunch.
Maybe one question.
So next speaker is Jilong Chen. He's working in Jun Song's group, and today he will tell us about possible applications of the Ihara setup function to graph clustering.
Yeah.
Which one the pointer are? Okay, yeah, sure. Okay. So, sorry about the confusion. Hi everyone, my name is Yidong Chen. I work with Professor Jun Song. So we are developing new methods, computational methods to study chromatin folding structures. So the new thing we use is something called IRZ functions. I'll tell you about what IRZ function is, and then this work is still preliminary, so I'll tell you what the difficulties we met, and then tell you the potential solutions that I have in mind right now. So first of all, the introduction. As we all know, the chromatin structure has a hierarchy of structures. So at the very bottom, around several kilobases of lengths, we have those looping structures. And these looping structures are structures that we want to study using these IRZ functions. Basically, we want to take the high-C data, reproduce a graph from the high-C data, and then study the graph structures by some concise statistics of those graphs. So as a picture, those are the looping structures. Those are the looping structures I'm talking about. So if we can turn, using high-C data, if we can turn these looping structures into a graph and then just study the graph computationally, we might be able to study the similarities and differences between those folding structures. So how do we turn the high-C data into a graph? First of all, I have to tell you what a graph is. So very concisely, a graph is a set of vertices and a set of edges. And this is a graph. These are the vertices, and these are the edges. So to construct a graph, I have to tell you what the vertices are. Also, I have to tell you how to connect those vertices, namely what are the edges. So the procedure of turning high-C data into a graph is like this. So we take the high-C contact matrix with resolution 5 kilobase, and then consider for each n, each of the location, consider locus n to n plus 49, which means I take the nearest 50 neighborhoods of n, and consider the submatrix of this region, Cij, whereas I and j bigger than n and smaller than n plus 49. And then arbitrarily fix a cutoff number. We don't consider all the contact. If the contact number is too small, we just throw it out. If the matrix entry Cij is larger than k, we retain that number and then construct a graph with 50 vertices. And then if Cij is larger than k, we connect vertex I with vertex j with an edge, and the edge has a weight of Cij. So this is the way we turn the high-C data into a graph. So as you can see, for each n, we have a graph of size 50. And then the goal is to use the I-Z functions I'll tell you about later on to study those graph structures. So high-C data resembles an ensemble average of contact frequency. Those neighborhood graphs I just constructed will represent on average, the local chromatin structures. So the important question now is what can we say about those chromatin structures from the graphical representations? This is where the I-Z function comes in. If we go back to the graph example, and I label the vertices in this way, this is vertex 1, 2, 3, and 4, and we can see that each graph contains some triangles. Like 1, 2, and 3, we have 2 triangles, 1 and 2. Also, we have some squares. 1, 2, 3, 4, you can see there are actually 4 squares that with vertices 1, 2, 3, 4 in this graph. So these are something called the local structures of the graphs. And if we can come up with some statistics to summarize those local structures, these are very useful in studying the chromatin structures from those graphs. And as you can imagine, if I have more complicated graphs, there are more structures like pentagons, hexagons, etc., etc. And as I said, these are crucial information about the graphs, which contains important biological information that we want to study. So we want to extract the number of triangles, squares, pentagons, etc., from this graph. So I-Z functions achieve just that. This is a function that is something called a generating function. It contains all the informations of the local structures into one big functions that we can use and compute. The coefficients of this function will be the number of triangles, squares, etc., etc. The good thing about I-Z function is if we want to compute these numbers, we don't have to do the computations separately. We can just do the computation at once because as a formula later on will show that computing I-Z functions amounts to computing the spectrum of a matrix. So the computation is very easy to achieve. So here's the definition. We have this function. This is the I-Z functions. U is the variable. M here would be the number of edges of the graph. You see, we have, as a polynomial, we have a degree 2 times m polynomial. And all the CNs will be the coefficients that contain the structure information. What do I mean by this? For example, the negative C3 is the number of triangles in your graph. Negative C4 would be the number of squares. Negative C5 would be number of pentagons, etc., etc. But these coefficients, if you want, you can think of them as a new kind of statistics of your graph. As a comparison, compare with the spectrum of the graph. If you have a graph, you can compute something called a spectrum of the graph, and then these will give you some information about the graph structures. But here we have something new. This is more intuitive, and as I'll tell you later on, this has a distinct advantage over considering spectrum of graph. So how do we compute the hierarchy functions? Obviously, we cannot just compute by using this definition, because if we want to do this, we might as well just count the number of squares, number of triangles, etc., etc. The good thing is that we have an alternative definition of a hierarchy function, which is the determinant of identity matrix minus u times t. T here is a 2m by 2m matrix, which we can compute directly from G. It's somewhat complicated, but it is doable, and then I have wrote an algorithm to do just that. So to compute hierarchy functions, we just have to compute the eigenvalues of t, which is easy to implement on a computer. As an example, or a sanity check of my algorithm, I did this graph image clustering problem, which I took 3 images of cups from the COIL database, and then tried to use my hierarchy function algorithm to cluster the 3 cups. First of all, I turned the cups into graphs by picking out the feature points, and then connect these feature points to turn them into a graph, and then compute the hierarchy functions, and then do the clustering. Keep in mind the COIL database has, each cup has 22 different, they took pictures of the same cup from 72 different angles, so we have 72 data points in the cluster. As you can see here, we have the 3 clusters. The blue dots would be the first cup, green dots second cup, red dots the third cup. You can see that these clusters clearly separate, which means the hierarchy function algorithm is working here. By the way, this is not original work. This is sanity check. People have done this in the computer literature. I just want to check if my algorithm works, and here this sanity check passes. Okay. So as I said, this work is still ongoing. I'll tell you what the obstacles are. The contact numbers gives weights to each edge in a graph. And it is believed that those contact numbers is a representation of physical distance between 2 chromatin loci. So we have to incorporate the information of contact numbers into our algorithm. But the algorithm I showed you that works, passes the sanity check, is an algorithm that works on the unweighted version of hierarchy functions. How to incorporate these contact numbers into the algorithm is a difficult question, and it's a question I'm still working on right now. So the goal is to use a version of weighted hierarchy functions to do the computation. I want to extend my algorithm to the weighted version, but it's not the straightforward application of the previous algorithm doesn't really work, because basically, if you have the weights, the information you extract from the matrix T that I showed you isn't clear. You can do the same computation, sure, but what the information you extract is complicated by the weights. So we have to find an interpretation of those computations to come up with useful information that we can use as a statistic of those weighted graphs. So yeah, this is what I'm doing right now. Find out quote-unquote physical interpretation of those weighted hierarchy functions, and then we can use the high-C matrix, contact matrix to produce similar heat maps that uses these weighted hierarchy functions. And then if I can achieve this, the further question is if we compare this heat map with the original heat map that people produce using high-C contact matrix, and then the spectrum of those high-C contact matrix. If we do the comparison, what biological information we can extract from those information. This is another step we want to do. So as a conclusion, hierarchy functions is a function that gives you a precise description of graph structures. We would like to develop new statistics from those hierarchy functions in order to classify the similarities and differences of chromatin folding structures. This work is ongoing and over and over again. So there are lots of questions, but we think this is going to work because, as I tell you about, the hierarchy function is a clear and concise summary of graph structures. With that, I think that's the end of my talk. I would like to take questions right now. Thank you.
I can just ask.
Yeah.
You, in one of your earlier slides, you talked about having some kind of cutoff k.
Yeah.
How do you choose what that value is?
So you can choose it arbitrarily, but the way I did it is I took, if you remember, I took 50 bins, right? I studied the statistic of those 50 bins. I studied extracted mean and medium. I think I took, I cut it off I took the medium because the distribution is highly skewed. I just, if it's less than medium, I cut it off. I just don't consider it. And then those two edges, those two vertices wouldn't be connected. Other questions? Yes.
So I don't know if I remember correctly, but can you extract like a number of triangles and squares instead of graph fashion?
Yes, you can, but if you do the computation, going from graph Laplacian to the number of triangles, it's not straightforward computation. So usually people would do.
Like, for example, take the cube of the graph Laplacian.
Yes, you take the trace and take the cube. You can get that, but if you want more information, for example, you want number of squares, number of pentagons, you have to do this computation over and over again, and then there is overcounting. If you do the multiplication too many times, there will be overcounting. For example, if you have you multiply your Laplacian 6 times, you'll get the number of 2 triangles, and then hexagons. This is the overcounting. And this is very complicated, but the hierarchy functions I have just defined will avoid these problems. So it's a very efficient way to extract the same information. Yeah. Other questions?
We have time for one more question.
How does the computational effort get? Because yes, you have redundancy problems.
Yeah.
Evaluating the determinant it takes longer than just doing the count.
Right. I cannot give you a quantitative way to study those because I haven't done a quantitative study of the computational complexity. But as is, the algorithm I've written down can be run on my laptop, and it's very fast. But that's just heuristic. I cannot give you. I haven't done the analysis yet. So. Yeah. But the way to turn from the graph to the matrix T, I showed you about. This is the most computationally intense part of this algorithm. But there is a way to do this step efficiently. Basically, you took your contact matrix, and then you considered a bunch of copies of those contact matrix. You took out the 0. If you have all 0 rows, all 0 columns, you throw this out, and then you build a larger matrix, and then you extract your L from this large matrix. This step is, I have to do some derivations, but this step can be done very fast. Yeah?
Can you give a sense of some biological interpretation that you can draw from this?
Yes, but the goal is to, as I have said, the goal is to study the similarity and difference between the folding structures. So we have those summaries of those little graphs, right? And then we can extract those summaries, like the number of triangles, number of squares, number of hexagons, turn them into a vector, and then just compare these two vectors as a comparison of the local graphs. And those would be a kind of way to extract the information, just to compare the local structures. If two, for example, along the chromatin, if 2 parts have similar vectors, we can say for sure that these 2 folding structures are similar in some sense. And if they're very different, they're different in some sense because they have different number of local structures. That's one way to extract biological information. The other thing I alluded about is that we can also build the heat map using these features of our graphs, and then compare our heat map with the heat map that people have produced using high-C contact matrix and the spectrum of the high-C contact matrix. compare these two heat maps, see what we can say about the difference, because they have to be different. But why are these differences? These are the questions I want to study later on. But the first step is to find out how to do the computation on weighted inheritance functions, as I have said.
Oh, that's it for the questions. Please another round of applause. So the next speaker is Surab Shukla from Paul Seven's group, and he will be talking about versatile force sensor for multiple motor cargo transport.
Hello? Is it audible? Okay.
Hi everybody, I am Saurabh from Selvin Group, and I am here to talk about my work on molecular motors. So molecular motors are a class of proteins in the cells which walk on cellular roads and which are responsible for cellular cargo transport from one place to another. There are three kinds: kinesin, dynein and myosin. Kinesin and dynein walks on microtubule, one type of cellular road. Myosin walks on actin. Here is the movie of dynein molecule walking on microtubule. Other motors also walk with slightly different mechanisms, very similarly. We have been interested in study of these proteins for quite a while now because they are very important for cellular function, especially in the brain when they are important for cell-to-cell communication, and there is evidence to suggest that malfunction in these motors and their auxiliary proteins such as tau are responsible for Alzheimer's disease. So that is why we are interested in delineating the molecular mechanisms of these motor proteins. So what happens in the cell? Cellular cargoes are transported by multiple motors. Several kinesins, several dyneins are present on one cargo, and they somehow coordinate together to get the cargo from its origin to its destination. Also, there are several microtubule associated proteins which act as a roadblock in this motion, and somehow these motors together, working together, overcome these roadblocks to ensure a smooth cargo transport. So primarily we are interested in two questions. One is how these motors coordinate with each other, that is how multiple motors work with each other, and how do they avoid these cellular roadblocks. So there are two ways of studying this problem. One is a classic walking assay where microtubule is immobilized on the surface and the individual motors are observed. Second way to study these motors is to flip this assay where these motor molecules are immobilized on the surface and the movement of moving microtubule is observed. So in these kinds of assays, one problem is that in the multiple motor scenario, it is not possible to see what these individual motors are doing, what their dynamics is. To study that, we developed a new kind of assay, which we call a force gliding assay. The kinesins are attached to the surface, but there is a slight modification. We insert a small piece of double-stranded DNA molecule between the kinesin and the surface, and this microtubule is moved. What this dsDNA piece does is it imparts some sort of flexibility to this kinesin molecule, and this kinesin molecule can fluctuate or displacement around its attachment point. So after doing these experiments, we discovered that kinesin can stay in two states. One of the two states, one is called driving kinesin and another is resisting. These names are chosen carefully based on their function, and I'll explain in the next slide how we differentiate between them and how we know their function. So I'll give you one example. These three dots represent three kinesin motors. They are driving a common microtubule. One thing to notice is that they are sufficiently far away from each other. The assay is designed such a way that they can be observed individually. So you can see that they are displacing around their original position, which is equilibrium position. So now we can track their displacements, and that displacement is correlated to their function. To go in the detail, if I focus on one kinesin molecule, let's assume the microtubule is moving in the forward direction. Initially, it is on the equilibrium position. In the middle panel, the kinesin displaces in the backward direction, that is in the opposite direction to the microtubule velocity. This is driving position. In this configuration, kinesin is actually generating the force and driving the cargo transport. It can also stay in resisting position when it has a positive displacement along the microtubule velocity, and in this case it resists the motion. So in our assay, it is possible now to delineate or differentiate these two states, which was never possible before in any of the assays previously. So what we do, we take hundreds of these movies, we track each kinesin, and we plot their displacements. For this case, we have plotted the displacement of kinesin 1, 2, and 3, and we have also in real time calculated how the cargo velocity is affected by their displacement. So I would draw your attention to kinesin 2. You see that initially the kinesin 2 has a negative displacement. That means it's in the driving position, and then it switches to the hindering position. That means that each kinesin is dynamic and it can give force or resist the motion. But you'll notice that when kinesin 2 goes to hindering or resisting position, another kinesin comes in and rescues the motion, and which basically explains why multiple motors are important to ensure the smooth cargo transport. And by this assay, we can actually see that happening in real time on a microscope slide. The questions which we try to answer with this statistics or the results of this assay are the following. First question we ask, what does kinesin like? Whether does it want to be in resisting state or driving state? So we do these, we analyze all these movies, and we come to a conclusion that the motion is positively biased towards positive direction because kinesin primarily likes to be in driving state. So with this assay, we can actually differentiate between the cases where one kinesin is driving the cargo, 2, 3, and up to 8. And we see in every configuration, almost 2/3 of kinesins are in driving position. Next question we ask is how the velocity of the cargo changes by increasing number of kinesins. With this assay, since we can see the number, we can deconstruct what happens when there is one kinesin driving the cargo, when there are 6 to 8 kinesins driving the cargo, and you can see the peak is almost invariable. The velocity does not change very much with increasing the number of kinesins. That actually settles a very important question in the field, whether the velocity is affected by the number of kinesins. Next, we also study the roadblocks in this assay. So what we do, we introduce some roadblocks as quantum dots on the microtubule, and you can imagine these kinesins will hit these roadblocks, and these roadblocks will try to stop these kinesins. And we exactly plot this velocity histograms in presence of roadblocks, and something very interesting happens here. You can see the velocity plot, the peak, this remains constant, but there is a significant peak at 0 velocity. That means a lot of kinesins are stuck when there are a lot of roadblock, especially where there are lower number of kinesins. When there is one kinesin, there is a lot of peak. And as we increase the number of kinesins, That means as number of kinesins driving the cargo increase, this peak disappears. That gives us a conclusive evidence that when there are more number of kinesins, despite of the presence of roadblocks, there is smooth cargo motion. And this basically gives us an indication or explains how, despite of presence of numerous roadblocks in the cell, how multiple motors are able to ensure the smooth cargo transport. We proposed a model based on this, but I will not show you a schematic, rather I would show you a movie. Let's say this is a panda which represents a kinesin moving alone, the microtubule. There is a certain velocity. He asks if my friends join me, and you see if there is increase in kinesin, the velocity does not change very much. So it's inconclusive. But the effect is prominent when there are roadblocks in the system, because he doesn't have enough force to overcome the roadblock, the microtubule gets stuck. But again, he calls upon his friends and they get a productive party together on the beach, and the roadblock is overcome. So this is one story which tells us the story of how multiple motors actually ensure the cargo transport. After this story get published, we ask ourselves whether we can actually go further and see or quantitate the force each kinesin is imparting on the microtubule. And it is an incredibly difficult problem to solve with the existing techniques. But we ask the question whether this dsDNA molecule can act as a force sensing molecule. Then we looked at the force versus extension of dsDNA. You can see it increases very gradually with extension, the force. And then at higher extension, it shoots up, which causes huge uncertainty in the measurement of force. Then we looked for other polymers and we zeroed in on single stranded DNA. This is the dsDNA plot and this is the single stranded DNA plot, and you can see the single stranded DNA force versus extension is gradual. It's has a moderate slope. It is approximately approaching to linearity, which gives it a huge force sensitivity, and that's what we chose for our essay. And then we focused our efforts to synthesize or purify this dsDNA molecule. For our essay to work, we had to get the ssDNA with dual labeled ssDNA. It was long. It was like 1200 base pair long. No company can make this for us, so it was huge effort, but we were able to synthesize and purify this DNA, which let us design this force sensor, dsDNA molecule, the motor proteins, and the microtubule goes. Its mechanism is very similar. We get the extension of the individual kinesin motor or dynein motor, while multiple of these motors are driving the cargo, and from these extensions we get the value of the force. We have experimentally verified it with our collaborators, Professor Jan Chamla's lab. I have that slide, but I will not be showing it. I have it in supplementary info, and we have some results. Now we can actually quantify the force of each kinesin motor while they are together carrying the cargo, and we see that the force is around 1.78 piconewton average force of individual kinesin motors. We can also quantify the total force on the microtubule by total number of kinesins which are driving the microtubule, and we can see the forces on the microtubule can go up to 10 piconewton, which is much higher than what is possibly can be producible by single kinesin, which is 7 piconewton. That basically gives us a conclusive evidence of additivity of forces in the multiple motor transport, and gives us hard numbers how much exactly the force is with real time. Now, to conclude, I have given you two stories. One, the mechanism of multimotor transport, and second, a design of a force sensor. And I'll give two snippets for the future directions of what our project is going. Our force sensor is versatile. We have shown it with kinesin, and it can be used with dynein as well as myosin, and it can be used in various configurations. We can use it with in presence of roadblocks, different kinds of roadblocks, tau, MAP1, MAP2, which are important in diseases. And we can also do this mixed motor assay where we can have kinesin and dynein working together on similar cargo, which is actually very similar to what happens in the cell. So we are very excited to do these experiments, and In conclusion, I'll like to thank my advisor Paul for being an excellent mentor, and my colleagues from Selvin Lab, Dr. Marco Chu, Dr. Marun Mehti, and I would like also to thank our very productive collaboration with Professor Jan Chemla's lab and Alice, who is here in the audience. Thank you so much for listening, and I'll be happy to take your questions.
Yeah, we have time for like 2 or 3 questions.
Yes.
So with the multiple kinesins doing better against the roadblock, are they just like what is the mechanism? Are they just pushing the roadblock out of the way?
So what happens is when there are multiple kinesins, there are 2 things happen. One, which we showed in the force sensor also, the force which is imparted to the microtubule which increases. So basically they push harder. And when there are multiple kinesins, what happens? Let's say one kinesin is stuck at roadblock, and other kinesins are pushing, which are not at the roadblock. That stuck kinesin, because of the force, detaches, and because there are other kinesins present in the system, they keep moving the microtubule. If there had been only one, the microtubule would have been lost, because there is only one, it detached and motion is gone. But there are others, the motion is continued, there is a smooth motion. And in the cell also there are multiple kinesins, one kinesin detaches, there are others which can actually keep carrying on the cargo. That cargo does not get lost in the cytoplasm. That's what we observed here. We can't observe it in the cell because they are so close to each other. Yes?
So is it correct that these microtubules have some helicity to them, and what role might that play?
So we know that kinesin, so there are 13 protofilaments to the microtubule, and there is very slight pitch, which is like few nanometers, not a lot. So we know the kinesin walks on the same protofilament. So when we are doing the kinesin experiment, that helicity will not make any difference. But we also know that dynein goes helically on the microtubule if we do the assay. So when we do the experiment with dynein, then it will come into play, but not with kinesin.
Have you systematically studied the spacing between the keys?
Yes, we have. So I didn't show the data, but this is the part of the paper we just published. When the spacing between the kinesin increases, the detachment rates of kinesin increase. But that doesn't matter because there are more kinesins on the cargo. So that's what this is one of the effects we got. But there are several other effects which can be derived.
So does that mean the closer is better for transporting?
So in cell, we can't control it. They are very close because imagine a sphere and there are 20 motors on same sphere and they are moving it. They are very close to each other in the cell. Here we can control it. In cell, we can't really. If there is extended cargo, like mitochondria, maybe, but of course, like spherical cargos like peroxisomes and endosomes, they're very close to each other, generally.
Thank you very much.
Thank you so much.
And now the last speaker of the day, David Bianchi from San Lupi Schulten's group. He will tell us about how to account for cell-to-cell variation in the small RNA mediated E. coli sugar shock response.
Can everyone hear me all right back there? All right, awesome. So first of all, I'd like to thank Luis for the introduction and the CPLC and the organizers for giving me the opportunity to talk today. And today I'd like to talk to you about how we can observe cell-to-cell variation in the E. coli sugar shock response. And this talk should hopefully dovetail really nicely with what Meng spoke about earlier about cell-to-cell variation. So in the Luthi-Schulten group, we focus on stochastic effects in cellular systems. And so like Meng pointed out before, there's many examples of the importance of cell-to-cell variation in biological systems, one being this experiment done by Elwitz in 2002, where they put a GFP protein promoter and RFP equidistant from the origin of replication. And what you'd naively expect is that if these were transcribed at the same rates, we'd see a bunch of yellow fluorescence here, but instead we can see a wide variation of colors. And this is really motivated frequently by low copy number species, such as genes or mRNA being present in the cell, and the reactions that they participate being characterized by this intrinsic noise. And this gives rise to variants of cell populations like we can see in this image here, and like we'll see in the sugar shock response later. So in the CPLC, we have a pretty big tradition of studying how cells respond to chemical environmental changes. And so one system, a chemical sensing system that this is really interesting in is the E. coli chemokinetic response, where the cell responds to the presence of attractants or repellents in motivating what direction it chooses to swim. And this work was done by Jan and Roshni. And just as a plug as well, Keith, who is an alumnus of the CPLC, will be giving a talk about the molecular details of this chemosensory array this afternoon for the CPLC seminar. And another area where we've studied this is some work I did in yeast, where we study how the cell responds to the presence of a new sugar, galactose, and how that gene regulatory network produces more of those green transporter proteins that you can see being produced on the exterior of the cell here when it's presented with that new sugar. And so how we do these simulations to capture this populational variability is by sampling from the chemical master equation, where we keep track of the number of different particles in the cell, whether that's mRNA, small RNA in this case, or proteins. And we keep track of transitions that change these particle numbers, whether that be a reaction like translation that will give us an extra protein, or mRNA decay that will decrease the number of RNA. And we've developed a software package, Lattice Microbes, that can input different spatial data like cryo-ET, or chemical data like in vitro kinetics or microscopy, like we'll see today, and enable us to use this in our workflow. And so the system we're going to talk about today is a project that was started several years ago in the CPLC, sugar shock response in E. coli. And so in that yeast example I showed earlier, the cell tries to produce more genetic material, use a genetic regulatory network to produce more material to metabolize the sugar. But what about the opposite case, in which too much of a sugar, like glucose or the glucoside analog alpha-MG that we use in this experiment, causes a big amount of chemical stress for the cell? You want to be able to dampen that down. And how that's done in this cell is a small RNA, SGRS, in the red here, or these black lines, blocks the translation of PTSG, these green wiggles. And PTSG is a transporter protein that brings glucose into the cell. So by blocking translation, the cell can reduce that chemical stress. And so just really quickly, how these siRNAs work is they're often small, often about a couple hundred nucleotides, and they'll bind by complementary base pairing to the RNA and including ribosomal binding sites, thereby reducing translation. And so some additional features we've added to this model to kind of account for this noise in the system that wasn't looked at previously is transcriptional regulation of the SGRS gene by an activator, SGRR. and then stabilization of this sRNA so that it is not degraded as quickly by the degradosome or RNA-seq, for example. And there'll be one other feature that Meng-Yu talked about earlier, gene replication, that also plays a very important role. And so, as I mentioned before, the additional features that we'll take into account are the genes for PTSG and SGRS replicating over this fast-growing E. coli cell cycle, transcriptional regulation, which has a significant effect on the noise, and explicit binding of the chaperone protein, HFQ, to stabilize the small RNA. And the experiments that we couple and use to inform our simulations are done in TJ Haas' lab at JHU, and we use SM fish where they hybridize these oligonucleotides to either the small RNA or the mRNA, and then they use storm microscopy to be able to analyze the different numbers of either PTSG in green here or SGRS in red from cell to cell. And these experiments give you a really great opportunity to see the variation in the response to this network across various cells. And so the final question that I kind of alluded to before that's very important is considering how replication occurs, especially in fast-growing cells. And so E. coli has often been measured to take about 40 minutes to replicate the genome. But the cells we're looking at here and many growth conditions that people work with double significantly faster than that, in this case 35 minutes. So the question then becomes, how do cells replicate their genetic information for daughter cells if these daughter cells are being generated faster than the genome can be copied? And the answer to this question is the fact that the E. coli cells, the daughter cells, are essentially born pregnant, or that is with multiple replication forks. And we can see this in this nice work done by Younger et al., where they label the replication forks with CK. And we can see that you can have as many as up to 4 replication forks in the growth conditions they're looking at. And so a really important effect this has in our stochastic simulations and in the data that you can see experimentally is that the number of gene copies you have, whether it's a lower number like 1 or a higher number like 2, affects the noise you see in how mRNA is generated, how bursty mRNA is generated throughout the different times of the cell cycle. And so we keep track of this in our simulations by sampling the percentage of cells with genes either in this lower gene copy state, where you may only have 1 or 2 replication forks, compared to cells that might have genes in a higher copy number state, where you'd have 3 or 4. And the thing that's really important to keep in mind when doing this sampling is where the gene's actually located on the circular chromosome. So in this example, the small RNA SGRS is located extremely close to the origin, and so you might expect more gene copies to be present throughout the cell cycle, while PTSG is located much farther, about 90 degrees away from the origin. And so, as I mentioned before, SGRS is likely to have a higher gene copy number over the course of these simulations. As we can see in this figure down here, there's more blue dashes representing SGRS than the orange dashes representing PTSG. And so by taking into account these factors, we can capture both the average behavior. The simulation here is in black and blue, is the interquartile range, and the experimental data from the STORM experiments is in magenta and red. And a better way to see this, especially when we're considering stochastic behaviors, is actually the distributions of both the sRNA and mRNA that are produced. And these are just kernel density estimates of those distributions that we see. And the really interesting thing you can see immediately from these figures, especially in the SGRS case, which is in higher copy numbers, is that the peak heights give you an idea of what those gene copy number states are in each of the cells. So these peaks at higher SGRS copy number may be cells that have more gene copies of SGRS relative to this higher peak at lower copy number. And so by pairing these single molecule experiments and stochastic simulations, we can get a very robust description of response to environmental stimuli. And in the future, what we'd like to do is account for the spatial effects in that target search of the small RNA for the mRNA, especially since recent work that Professor Senjin Kim in CPLC has done has suggested that the degradosome is located primarily at the exterior of the cell. And so that could affect that diffusion time for those two particles to find each other. So I'd like to thank Professor Luthi Schulten, who directed this research, Troy Breyer, who from the Luthi Schulten group, who worked on me in this computationally in this project, Professor Carrie Vanderpool in MCV, and Dr. Shofi Azam, who were responsible for constructing the various bacterial strains and doing certain knockout experiments, and Professor TJ Ha and Anas Supadar, who were responsible for the SM fish and storm experiments in this study. So thank you very much for your attention.
David, so following up on what you said about the spatial aspect, so even before any of the work, the degradosome, remember one of the striking features of these data was that some of the mRNAs are localized in a helical pattern of the membrane.
Yes.
So the fact that your model does such a good job of reproducing the data without thinking of space either in terms of RNA localization or RNA localization, does that suggest to you that these effects are actually not important, or just the parameters manage to sort of compensate for that?
So I guess my response to that would be that especially the fact that at these later times, the SDRS is in such high copy number that kind of if there's some stochastic effects of those search kinetics of that spatial localization, that probably gets washed out by there being higher copy number of the small RNA that's able to find and bind to PTSG. And then the other interesting point in that, I know there's some studies about that chaperone protein, if I just go back here, HFQ, that suggests that it plays a role in establishing contact between the small RNA and the sRNA as well at the ribosome.
But just to follow up,
yeah.
In my mind, there's always, and that sort of seems to often happen in biology, that we can reproduce the data with a model that ignores important features, which tells us that a model that fits that does not at all tell us it is sufficient or correct, like probably.
Sure.
Any other questions? I was wondering, like, besides transcription and transcriptional silencing and gene duplication, do you think it's important to consider other processes?
So I would say in this system, why we primarily focus on those processes, kind of going off of some things that Menu mentioned in his talk as well, is that these are the particles in this species that are present in the lowest copy number and are most likely to exhibit that bursty behavior that's really going to lead to cell-to-cell variation. There are certain other proteins that are involved in this stress response network, but they're often present at a much higher copy number.
So I think that's it. Yep. Thank you, David.
We're about to do a photo with all the presenters that are still here. I think some have already left, but yeah.
All the presenters, can you please come to the front? I'd like to take a picture of everyone. And are we voting now?
Yeah. So
where do we put the
okay, that's right.
forms when we're done voting?
I guess we can just also have them on that table in the corner on the other side.
So please fill out a voting form and just stick it. Janice, can you make where Troy has Troy's making the pile. Thank you, Troy.
All right, and this is not going to work out. Can we turn it off, or should we choose a different location, or what?
We can just turn it off.
Yeah, the poster. That would be great, actually.
Charlene.
Do we have the poster? Yes.
Seminar today is in 144, right?
Yes, same place. Was there a...
It just wasn't on the website. Someone was asking me, and I just wanted
On
to
our
confirm.
website.
Okay,
Mhm,
okay.
but
They did.
We have a lot of other stuff on our
Okay,
website.
yeah. I just wanted to make sure.
Things going
Yeah.
on. What can I say?
Stochastic was asking me, you guys can all vote.
There we go. Yeah, just click on that thing. There you go. That's fine, right? Is that okay? Or can we make that big? Full screen. And
Yeah, here we go. Here we go.
there we go. Beautiful. Good. That's fine. Yeah, nice. Okay. Still a little weird with, yeah, that's good. Yeah, just don't stand right. Mayank, you can stand right there. Just when it gets to the white part, that's weird. Okay. And you might have to kind of like, I don't know. Do you want to put a chair?
I'll let just me.
No, you guys should be in it.
Yeah. Okay.
So let's see if you sit there. Why doesn't somebody sit there? And let's see.
Okay, all right.
Just anybody who wants to sit. I want to just see that the light is not in your face. If the light's in your face, then it's a no. Yeah, that's good actually. Okay. Do you want to sit next to it?
No.
Okay. Let's
It's an empty chair.
David just vibe.
Yeah, sit down. Look like we are the director. Looks like we are the director.
If we have
right?
one more, if we have one more person, then
You can share Charlie over sit here. Charlie, you should sit here.
No, I'm not going to sit anything. Okay, so maybe, yeah, somebody who's on the edge. Well, there are too many
Whoever's the
people over
shortest
here.
man can.
Yeah, David, good. Okay, so now scooch together. Thank you very much.
Yeah, we got two Davids. We got a David sandwich.
Oh yeah, there are too many Davids. Yeah. We'll call one of you Dave. All right, 1, 2, and say I don't know. CPLC. Okay, I think that's good. I think it's good.
Yeah. Go get some food.
Do we get to vote or no?
Yeah, you guys can vote.
Everybody can vote.
Thank you for
Yeah, so I don't know if some of the emails missed you or. Are you not on the CPLC mailing list?
You're on. Yeah.
Because one of them is
But yeah, I think it was just a week before this. I was wondering which song ended up. Of course, it's not.
Okay. It doesn't really matter what they've done in the past.
Okay,
Yeah? Well done. I like to see your edits that you made too. And actually, you caught it this time, and I forgot to mention it. Yesterday you said,
per second. Yeah. Well, we can do one for each and then a fourth that has all three.
Yeah, because probably the one that has all three that's probably the most. So hold on just a second. Let me see if there's a bag that you have to. Yeah, I have to go. Well, in fact, you have to
yeah. I think I see that a couple times now. But yeah, well done.